everybody. We would like to warmly welcome everybody present here today at the Natural Science Baltic Conference. It is our absolute pleasure to host you at this significant online event, and we truly hope that the NSBC will be just as successful as the previous edition of the conference, which is the Baltic Chemistry Conference. Now, just quick notes to start it off. As soon as the presentation moves. There we go. It is important to note that our current conference is interdisciplinary. It combines 3 different fields, which is chemistry, biology, and physics. It is also international. We are absolutely pleased to welcome speakers from 8 different countries and 27 different scientific centers from all around the world. Additionally, due to the use of online communication tools, the conference will always remain free of charge. Now, the main organizers of the conference are all from the University of Gdańsk. This is the Student Science Club for Chemistry, the Doctoral Student Council, the Chemistry Student Council, as well as the Center for Student and Doctoral Activities. You can, of course, view the full organizational committee on our website. The event is also under the patronage of Fahrenheit Universities, Polish Chemistry, the Polish Chamber of Chemical Industry, the National Representation of Doctoral Candidates, the Polish Academy of Sciences in Gdańsk, the Dean of the Faculty of Chemistry at the University of Gdańsk, and the honorary patronage of the Rector of the University of Gdańsk. It is also supported by the Pomeranian Voivode and the agreement of the Academic Technology Transfer Centers, as well as Edoktorant and Teraz Środowisko. Additionally, we would also like to thank the Scientific Committee for its invaluable scientific contribution towards this conference. In this edition, the members of the Scientific Committee are from the University of Gdańsk, Professor Anna Axman, Professor Marcin Marciniak, Dr Elżbieta Adamska, Dr Małgorzata Kropusta, Dr Magda Dyńska, Dr Wojciech Snoch, Dr Emilia Gontarek-Castro, Dr Anna Pancielejko, Dr Katarzyna Węgrzyn, and Dr Wojciech Siwiak, who is also a representative of the International Center for Cancer Vaccine Science. From the Jagiellonian University, we have Dr Maria Madej and Dr Dawid Wnuk. From the Warsaw University of Life Sciences, Professor Katarzyna Wiktorska, Dr Agata Kuczko. Additionally, from the Jasiej Tech University, we have Professor Henry Pinto. From the University of St. Andrews, Dr Amod Desai. From the Polish Academy of Sciences, Dr Monika Hejna, and from the University of Opole, Dr Dawid Zich. And lastly, we have from the Gdańsk University of Technology, Dr Maria Kochaniec. We would also like to welcome and express our deep gratitude for our special guests who will be presenting keynotes. Dr. Manuel Banzhaf from the University of Newcastle will be delivering a lecture today on using systems microbiology to combat infectious diseases. Tomorrow we will hear from Dr. Joanna Brzeźdżoń from the University of Gdańsk, who will be presenting a lecture on self-healing polymer synthesis application and future research directions. We would also like to warmly welcome the speakers of the presentations and posters. Please remember that you do have a 13-minute time session for each presentation, followed by a 2-minute discussion period. The poster sessions will take place on between 3:45 p.m. and 4:45 p.m. UK time in special channels under the poster session tab, which can be found on the conference website. And also, we would like to invite you to regularly check up on the schedule of the conference should any issues such as delays occur. Now, if you encounter any issues at all, please contact us at the provided email here. It's the nsbc@studms@ug.edu.pl. We will try and solve any issues as quickly as possible. And lastly, thank you once again, and we invite you we wish you to have a wonderful conference. We will begin this conference with the inaugural lecture of Dr. Manuel Banzhaf from the University of Newcastle, after which the first session of oral communications will begin. Dr. Banzhaf, are you here with us? There you are. I cannot hear you right now. We can see you, however.
So, okay, can everyone hear me?
Yes, now I can hear you. Sorry, I just want to make sure. Did I pronounce your surname correct? Is it Banzhaf?
Yeah, that's all right. I'm not picky. It's an odd name.
Okay, so the floor is yours.
I think I need to share my screen. Can I do this?
Yes, I've already stopped sharing mine, so you should be able to share yours.
I have we need permission to share screen.
Okay, let me quickly communicate this with my team and we'll see what we can do. Yeah, it would appear that the current settings of the meeting will not allow you to share your screen. We are trying to change the settings as quickly as possible, so hopefully we'll have this sorted soon.
Okay, thanks.
Maybe in order to not waste so much time on the settings, we would perhaps ask you to send the presentation through to us through email, and maybe one of us could simply share our screen, and you could just simply tell us when to go on to the next slide if you're all right with that.
Okay, give me a second.
Dr. Banzhaf, are you still here with us? Because you seem to have disappeared from the screen. We tried changing the settings, so if you want to try sharing your screen once again, perhaps it will work now.
Okay, give me a second. I think also yeah, I'm about to send it. Okay, does it work?
Yes, we can see your screen now.
Okay, perfect. Good. Okay, so hopefully you see my. God damn it. Okay, can you see my presentation now?
Yes, we can see your presentation. We can also see you and hear you clearly, so everything's
Oh,
good.
okay. So now after, it's a good reminder for conferences and things, technique always is an issue. So I'll try to, make it a bit quicker and so like that we still stay on time. So thanks again for the great pleasure for to speak here. So like what my lab is doing is we use systems microbiology to combat infection disease. So from your lab life, you know that you always work on interesting phenotypes, but to truly understand them and study them, you need to go to the genotype. And finding this like phenotype-genotype relationship can be often like finding a needle in the haystack and so. And systems microbiology can solve these problems because there we can work with like arrayed and pooled libraries. We can use robotics to do many experiments at the same time. We have very interesting like high throughput like setups and readouts and so like where we can look for fitness, we can sequence, we can image things. Then we can take our data, we do data analysis, try to describe everything like in a network based on correlations, and then we still pick out the most interesting hypotheses and we study them in the lab to do the molecular mechanism. That's what most people are interested in. So for this talk, I show you a bit like what we can do with high throughput approaches using arrayed libraries, and by that I mean that when we look at bacterial strain, but the same concept would also be for mammalian cells, and so like it's just that we know their genotypes. So here we have like a mutant cells, so for bacteria, so this would be now an AMI-A mutant, and it's the names doesn't matter, so we just pretty much know their genetic background. So what you can do then, and that's called like a chemical genomics, you have your mutants with a known phenotype and you can just put a drug and a stress on it. And that can be anything, you know, from like putting more salt in your media or putting to an antibiotic. And what you get there is like for your drug, you get a genotypic, a phenotype, genotype observation to your known mutant. And you know their name, like this is like A, this is E. And because that works in principle, what you can do, you can just do more of it and so like. And then what you get is like an epistasis map, a stress response map, where you can track the response to a lot of drugs for one gene and so like. And at that point, I have to introduce you to the concept of functional clustering. So if you think about a car, if I take away your wheels, your car will not be able to drive. But same is true also if I take away your engine. So like, because both are needed or essential for motility. And the same is true for a lot of like functions in the cells, like metabolic pathways, protein complexes. They all need each other to fulfill the function. So for the cell, it doesn't matter too much, like here in the example for the efflux pump, if the function of like the gene 1 is perturbed or the gene 3, so the pump doesn't work and so like. So you can use this now and link genes by their phenotypic signature to functions. So if we again like look at gene A and we look just through the signature, like if it grows better or worse in all the different drugs, and we did this like now for a lot of the very similar like mutants in for example, in a single deletion mutant library, can ask the question, so like who are the friends of gene A? And then bioinformatically, we just look for the correlation. So how similar are the responses? We see that gene F and gene I and so like are very similar. And that helps us then in the real world when we do the biology to reconstitute this function, like in this case here, the efflux pump as an example. So you can also find things that are not 100% correlated, but they're still correlated enough to be interesting, and sometimes only in some condition. And to go back to the car examples, so like some car examples are not essential for motility, but helpful. For example, car lights. So if you think about now, we would screen lots of conditions where you drive, and then likely motility and car lights in all conditions where it's dark and where visibility matters, and so like you would see a correlation between having headlights and motility. And the concept there is relatively similar. So what we do in our lab, so like we have a lot of these chemical genomics projects. So what I show you today is our work on the Pseudomonas aeruginosa chemical genomics, but there's pretty much a bunch of other bacteria species we currently work on. And you see more pictures later in the presentation. So it looks like this. So this is like a plate where you have 1,536 different bacteria on it, and we all know their genotype. And then they're like these little dots and so like, and this is like where we need a bigger format for Mycobacterium bovis BCG. So this is the vaccine strain of tuberculosis, and they're just a bit more complicated to grow because it takes 4 weeks to grow, takes a day to grow. So pretty much for each of these different strains, we still have to optimize it to make it work for strains, because the reason why we have so diverse bacteria is that they're also really diverse to work with. Anyhow, so let's directly go to Pseudomonas. So we have Pseudomonas is a gram-negative, 5,000 to 5,000, 6,000 open reading frames. So like genes, is facultative anaerobe, it's versatile metabolites, and is a quorum sensing model organism. So we'll probably read some papers about that. People use that to study quorum sensing. It's also really a model for biofilm formation. It's an opportunistic pathogen, and for me a bit relevant is like it's resistant to most antibiotics. So what we can do, we can take this pinning robot, which is really nothing else than a very expensive stamp, and then put the drugs and stresses directly in the agar. We just then overlay and stamp this library where, again, like each little dot is a different experiment, and we just add 200 unique drugs and stresses to pretty much emulate the various like lifestyle situation the bacteria can get into. So then we have to make sense out of this like images. So like how we did that is so like we developed our own image analysis software. So this is called Iris. It's like open source. You can use it for free. And what it does, it just takes the picture when we take that in the lab. And so like it can just segment it back pretty much to these like single tile, because each single tile is an experiment. And then we can look at like fitness, where we just count up the pixel sizes. We can look at color, because with image analyze we can understand how colorful something is. We can even, you know, look at morphology and give a score based on the degree of wrinkliness. We can look for sporulation, CPRG, and a couple of other standard like lab dice people use. And at the end of the day, it's an Excel file. So when you ever do something in the lab, it always, you know, somehow results to that you have an Excel file and so like. Now a bit like how we score that, and so like how we create this fitness scores, because there is a little bit more thought behind that than you think, because normally in the lab you would intrinsically always compare things just to the wild type like permutation and so like, and see the difference. But that's problematic if you have this single deletion libraries where you have one strain and you knock out pretty much all the open reading streams that you can knock out, because they all start from a different base fitness phenotype. If we look at fitness, so here we have like gene A, gene B, gene X, and this is like on your no stress condition, and there's already a big difference. So you cannot really just compare them to wild type, because your dynamic range from something that is very sick to wild type, even comparing these like B and X to each other and so like, it's just really unfair and skewed and so. So what we do then, we just compare them to each other. So how that works, and so like we now take here again gene X, and then we just screen that in all the conditions you can think of. So heat job, and 9-medium, ampicillin, cefzolidin, and anaerobic growth. And then just calculate a control size, because our null hypothesis is we work with non-essential genes, because otherwise we couldn't have knocked them out in the first place. And non-essential genes are normally only important in condition they matter. So like if you're a salt pump, then probably you're important in any salt condition. If you're a penicillin binding protein, then you're important in anything in envelope biogenesis. If you're important in UV stress, then you should have a function in all the UV stresses and so like. So if you take a very diverse set of conditions, they should only light up and so like where they have the function, and that's actually the whole purpose of this assay. So like that's why you would expect that if you screen 300 conditions, they should only be active in a way in 5 conditions. So to have like a normal distribution. So that's why you can just add up all the values and make a control size, because your null hypothesis is that in most conditions your cells should not be responsive. And then take this control size, go back to the data, and then see in a way like in what conditions your cells are responsive based on your control size. So in a way like you simulate their own wild type, which is much more powerful, because it allows you to see like the differences even for a mutant that has a very low base fitness and so. So and out of this and so like you can just make a score. So if it's green, then it doesn't grow, and if it's red, it grows better. Okay, so we used to do that with a MATLAB script, but the problem there was this MATLAB script was developed 2007 or so, and it was written by a PhD student. No offense against PhD students, but it was pretty much from a self-taught bioinformatician, and it was more or less like a black box, and we could only run it if we simulate a Windows, I think 2000 machine, and it needed to use MATLAB 8 or so like, and currently we're I think at MATLAB 25 and so like. So and this is a problem with old software, if it's not continuously maintained and so like, it really ages well. But because we do a lot of chemical genomics in our lab, so what we decided to update the software with new feature and bring it in Python and just make it accessible for maybe the next 20 years, and that software is called ChemGapt. And what it does, and so like you take the images and then we use iris to get the numbers, but then with the numbers, and so like we can do now the auto row normalization, because bacteria here, you can see that a bit on this picture, they grow bigger on the outer rows. Makes also sense because there is less competition here on the side, but that would skew your data. So like we have an algorithm for that. Then we take this experiments all on different days, but now you hopefully understand that we always compare each strain of each other, but you know, like then you need data from a lot of different experiments from different days, and to make that comparable, so like we normalize them, so like that also what the software can do. And then we have a lot of quality control tests, and so like where you can just see like reasonable thinking in a way like that you can fish out things that are a bit odd. So I'll just explain here the Mann-Whitney test, and so like, so if you just have a normal plate, so you can ask and just take the numbers there. So again, you take your Excel sheet and you have your replicates, so because we always do replicates, as you all should do. So like, so here we have replicate A, B, C, and then if you have your Excel sheet, your numbers, you just ask the question, how similar are the numbers from A to B and A to C and A to D? And you can see for this example here, A, B, C are pretty much similar, so they're certainly the same things, but then D is the outlier because it's not similar to all the three guys. And this is a classical case of someone mislabeling a plate. And if we screen like 30,000 plates, and so like, trust me, that happens more often than you wish for. And so that's why it's quite nice with the software, because it will not change your data, and so like you just run it through, but then using these different tests, and so like you just get a readout and again an Excel sheet, and so like it tells you like these conditions, have a look, and then you can decide. And a lot of times in a case like that, you will be even able to fix it and just like rename then the file, and so like, and then it makes sense. So, but what that brings us is pretty much to this like chemical genomics data set I hyped up. So like what we have now here, it's all our deletion mutants for Pseudomonas, and then we have our drugs and stresses, and then we can again use this car example to really see what things cluster, and we can look in these clusters and then, you know, look for validation and do hypothesis-driven biology. So this is again the efflux pump. So in Pseudomonas, that's like MaxAB and OrbM, and you can see that that naturally clusters. It's sensitive to a bunch of things, and so like, so this is was already known, but known biology helps us to validate our screen. That's why I don't really spend too many words on this. Here's something new, and that's not now on a metabolic pathway, that we not only talk about efflux pumps. So here's the biotin synthesis class, where you can see like there's all the genes involved in it, where we have mutants, they cluster nicely, and that works with a human targeted drug. So like, I cannot reveal the name because currently we work with a pharmaceutical company and they make you sign stuff. Anyhow, so for the talk of the time for the talk, we just called it HTD, like human targeted drug, and we had like 2 of the concentrations here, and you can see with green it's like that they cannot grow. So when you don't have biotin and you add this drug on it, and so like they just simply die, which is very interesting because Pseudomonas is a really nasty pathogen, so we're always looking for things that can kill it. So what you then do is like you replicate that. So this is pretty much all the strains and the wild type with very little biotin, and so like, and this is pretty much physiologically relevant. And then if we add this, you know, drug on it, and so you can see that they pretty much die, and so like, and this is at the case where there's still a bit of biotin around, and so. And then when we add more biotin, you can see the wild type growth doesn't really change. So like there's no growth advantage. So like if they can, because they can make their own biotin, they don't need it like as fed by them. But then if you add a lot of biotin and add the drug, you can see you can completely complement that. So it's really like this combination between not having enough biotin and the drug. So then we looked into the biotin operon structure of Pseudomonas and already saw that it's like very different. compared to a lot of other of the enterobacteria and so. And this rearrangement already could tell you that biotin seems to be much more important for Pseudomonas, and no surprise there. This is also was confirmed by another lab that biotin and Klebsiella pneumoniae, Acinetobacter, they need much more biotin for survival, and that also makes sense because bioinformatically they just use a lot more biotin-dependent genes because biotin is a cofactor, and E. coli has one, and Acinetobacter and Pseudomonas just used many more. So like that kind of, the genetics already tell you biotin seems to be very important. Then, because I'm the man of the libraries and have all these single deletion libraries in my freezer, so we tried the same experiment also with biotin mutants for Acinetobacter, but as you can see here, there's pretty much no effect. So whatever we found is like very specific to Pseudomonas. So what we can do then is like we can do survival assays, so we can add the Pseudomonas in C. elegans, which is a very cheap and ethically okay animal model. So we just add them, the Pseudomonas, for 2 hours, and then we just add the drug as a treatment. And this is now just the wild type, and you can see with increasing amount of drug, and so like nothing happens, so all the worms die. But then if you take a biotin mutant, so they cannot produce biotin, so like the killing is much delayed, and so like they die at some point anyway, so it's kind of like a cure, and so like. So the summary of that is, so like we know that Pseudomonas has more biotin. This human targeted drug, and so like kills Pseudomonas in the presence of physiological levels of biotin. It wouldn't, for the lack of the better word, I say treat, and so it's really an early animal model, so like don't take that too far, but you can at least like extend the lifespan if you add this HTD to C. elegans, and then now currently we try to screen for an inhibitor. We try to do the same thing in mice, and we also study to understand the whole thing from the worm side, and so like, and that's together like with mainly Felipe Cabrera from University Cologne. So the next thing we wanted to do is like then to compare this whole data sets between two chemical genomics data sets between two strains, because we really wanted to understand how valid our data sets are in itself, and so like, and that's why we repeated everything I told you before with already two strains, PO14 and PO1. There are two libraries that are available. They're both wound isolases, still like brothers and sisters. They're genetically very similar, so the difference is like 234 genes like for that side, and then 400 for this, but they're mostly on genomic islands, and you can repurpose these islands and they still have a different phenotype. And the idea is to try to understand if it's the gene content that makes you unique, or is it how you use it. And so like, so it's pretty much a question in a way like how they repurpose their genes. So we screened everything in the same concept. So that's pretty much like now another viewpoint of what I already told you before. So these are all the different antibiotics, and because of the quality control, they're not exactly matching, because sometimes the condition just looks really awful of one of the strain, then we have to remove it. But overall, you hopefully appreciated that it's more or less, and so like a very similar data set for both libraries. We looked at the reproducibility for both libraries, and that's again like using ChemGap, just seeing how many dots have the same, response over the replicates. And there we are at something R values around 0.7, which for these kind of screens, because if you think about, these are like tens of millions data points, and so like it's actually quite good, and so like, so you wouldn't expect that to be like a 100%, and so like, because there's a lot of noise how we do the experiments. And then another question we can ask, how many mutants can show a significant phenotype? So you can take each mutant, again, go back to the phenotypic signature with a car example, and you just ask them it at least on one of the 200 conditions we screened, they are significant compared to all the other strains on the screen. So like they're like in the top 10%, like the front runners, and so like that either grow slower or bigger, and so. And there we can see with both libraries, we capture roughly half of the strains, and so like, which makes sense if I shall, because I will show you later. Here we really only look for fitness, and if you think about all the genes you have in a bacteria, they're all not for fitness related, so you would not expect that you get phenotypes for all the genes, because we can only look for fitness in that screen. So like everything that is not fitness related, you just wouldn't capture because you don't have the right readout for it. So the point from all that is like it's relatively similar. We can look at like multiple stress response genes and so like, and their response distribution. So like this is very niche in a way. Like, so we look at the genes that have the most phenotypes. So they have like 15 phenotypes, and then see where they respond. And you can see here it already diverges. But I wouldn't say that, you know, it's now a clear tell what is going on. This is more interesting. So like, because they're brothers and sisters and they're 95 identical, what you can ask is by doing one of these, you know, dimensional tests, and so like how much they overlay. So like here in pink, you see like all the response of the screen for PA14, and in blue you have it for PO1. And if they would really have the same response, if this is now the homologs paired to each other, so like let's say gene A in 14 and GA in 01, and so like you would expect normally if they're the same and everything works the same, that they're pretty much like are very close to each other. But that seems not to be the case, and so like if you do this, which was a bit surprising. But interesting at the same time, and so like, and we looked at one of these examples. So here is you know, like it's about stringent response, which is super complicated, and I don't even get it. So like I will be very light on the detail. If someone works on stringent response, please don't ask me a question, because if I even hear PPG, PP, I just like, it's so complicated. But anyway, so Pseudomonas kind of uses this, you know, Alamon metabolism in the cell to make a lot of decision about life, and so like, and these levels are constantly built up and degraded. And one of these hydrolases is this PASHA, that pretty much hydrolyzes that to this GMP form. And so like there are many more, and so like as I said, it's and I'm not saying that as a joke, and so like it's really highly complicated stringent response in Pseudomonas. And the interesting part here is like if you look at this hydrolase, Alamon hydrolase, and so like in the response to like sulfo drugs, and so like there is nothing on the PO1, but it's a really very steep in a way, like they cannot survive in a way like in PO14. And then we went and we made actually clean knockout just to make sure, and so like that everything, no other point mutation, we sequenced everything, and so like, and we can really see that we even see a dose response. And overall, that made us think, and so like then when I present you now this chemical genomics data sets as the shit, and really cool to you know, get all the phenotype genotype relationship for your work, we really have to be careful, because maybe they're not so universal, especially for very versatile bacteria such as Pseudomonas. That doesn't mean that my PA14 data set is by any stretch of imagination wrong if you're interested in PA14, but PA14 and PA1, because Pseudomonas is such a variable and you know, grows in so many different host environments, and so like even though they have very similar genes, and so like they might use them very differently, and so like, and that's something I'm very interested in, want to study a bit more, and so like about this gene rewiring and gene repurposing. Okay, so I think I still have like a few minutes. So we can also look at non-FITRES related phenotypes such as biofilm. And you can see here from these plates that if you only look at growth, it's the same plate as with and without perturbation. They look very similar for growth. That wouldn't be significant. But if I add now a biofilm dye, you can see they're red, so they form biofilm. So by just adding this biofilm dye, we get 2 for 1. We get just a different readout. We can also look into like the wrinkliness, and so like, and because our software can analyze that, so what we then did is like a wrinkliness screen. So we took we reduced the format a bit and just made a time course. So this is now this time course. This is still Pseudomonas and so on. We can just follow it over time and can really appreciate that you get all these wrinkles, which is cool biology. So now you again can look at their genotypes and their phenotypes and then try to understand what genes are responsible for bioformation. What is also really cool is what our software can do. So this is all one replicate A, B, C, D, and they're all done over different times. There's probably a year apart the screens, and you can really appreciate that they always get a relatively similar shape and form and so they look like brothers and sisters. So which means the genes they have somehow make it happen that they look similar, a bit you look a bit similar like your parents and so which I found fascinating because I really thought there's much more variance in between and so. So what we then can do, we can look at the size, the morphology, and the color intensity, and we can kind of bin them and look at, because it's a time course, you look at the trajectory and so and you can see how they correlate. So this is now a bit like the machine learning part of that. And then what we can do, we can cluster them based on the bins we do, and then we can go term enrichment for the different clusters. And then now we try to figure out if we can look at the cell and then just by the look say okay, it will likely, if they grow like this and so then likely they're, the gene deletion is responsible for, flagellar assembly, metabolism, because it seems like depending where you perturb, you look a bit similar and so which is cool, but we just started in that and so and this is our image analysis machine learning project at the moment and so I don't know, it's let's see how we go. Ask me in 2 years again and so if that's possible or if it doesn't work. And then as a last side note, what I showed you so far, we did that all with tamed lab spec, tamed lab pets, but if you really wanna combat antimicrobial resistance, you have to really take into account the true wild type, and we have to do this with real lab strains, taking the pan genome in the hospital into account. And we teamed up with Jay Hinton, and he just sent us last week 5,000 sequenced Salmonella strains, where we have then not single deletion, we have the full Salmonella strains out of the clinic, all sequenced. And now we want to use the same phenotypic assays like fitness, biofilm, whatever we can do also with collaborators to really try to find point mutation or genetic traits that they can link that to phenotypes, and then hopefully that will allow us to find marker genes or regions that can predict the outcome of antimicrobial therapies. Because in principle, if you think about it now with sequencing being so quick and so you could sequence a patient probably within 2-3 hours in the hospital. You could have your nanopore reads and so and if that would already be good enough to predict which antibiotics they're resistant to, then that would be a major breakthrough for medicine. If that's possible, don't know. It's challenging because bacteria adapt so quickly, but you just got to try and see how far you get with your basic research. And with that, thank you for the attention. I'm a guy sitting in the office all day, so the real applaud goes to Jack and Hannah, which I presented most of their work, and thanks for all my funder that helped me to keep the lights on. And I'm happy to take your questions.
Yeah, thank you very much for that. So as Dr. Banzhaf already mentioned, we open to taking questions. If you have a question, just please raise your hand and we will call you out. Dominika, are you here with us? Can you hear me?
Yes, I am. I have one question. What are the main challenges of using bacterial libraries in antibiotic resistance research?
The main advantage or challenge?
Challenge.
So what I presented is not per se a drug screen, because I hope it came a bit clear that if you now take a pathogen, like Pseudomonas, and you really knock out every gene, so like you have Pseudomonas has around 5,000 genes, and I really have made this library, and we have like 4,800 different. So there are some that are essential genes, you just simply cannot get them. So you get all the accessory genes, and so what that helps you is like to create this matrix of where you have like a mutant and you have the phenotype. And that just helps researchers all over the world. So like if you work on Pseudomonas, you could contact me, said like I work on PA, the number, and you have no clue what it does. We can look back in this catalog, that's why it's called systems biology. And then I can tell you like, oh, it clusters with these 3 genes and maybe has a function together. And this additional knowledge just helps you a lot to understand mode of action studies, to understand how antimicrobial resistance occur, because you see like if it clusters with an efflux pump. So it's more like cataloging, because if you think about and you heard probably a lot about sequencing, and now everything is sequenced. We have like the sequence information from so many bacteria, but if you look actually in the sequencing, half of it or even more is not annotated. And you cannot use sequencing data if nothing is annotated. And what we do is with these assays is like we annotate everything, because at least we can give it a function, and there are data sets in which we create where you can see like, oh, this homolog or that, and so like that is sick in these conditions, and these are the friends, and so like. So in that sense, indirectly, and so like it's very helpful for antimicrobial research. It's not a drug screen, and I think if you would want to really address antimicrobial resistance, screen or mode of action, you need to do gain of function assays, and so like. So that's much more potent, but we also do this in the lab, and so like, but this is really like finding, caring for the orphan genes. Someone has to, so like we need to give them functions, and then these functions will help us all to better understand the biology of a bug, and if you understand the biology, then it's easier to combat antimicrobial resistance.
Thank you very much.
Thank you for the question. We also have a question from Professor Wiktorska.
Hello. First of all, thank you very much for a very interesting lecture. My question is, because I work on mammalian cells, is it possible, or do you think you can adjust this method for other organisms, like mammalian cells, for example?
Yes, and it's already been done. What we will get soon in Newcastle is Wellcome. A big funder in the UK, they developed a single deletion library, lentiviral library for mammalian cells. And I think in a few months I should have that. And there we have like then all the 30,000 lentiviral, and we can either mix them in a pool and then do a pooled assay, or we can have like any cell line or any human cell line you want, and so like, and we add the lentiviral in the land. In the well, and then you could add a pathogen or you could add a drug on it, and so like, and so this is available. In fact, normally the mammalian cells and the yeast cells are always far more advanced than what we do in microbiology. So also this, what I've just presented you, so like it's been done in yeast since the 2000s. It's just like was very slow to find someone, in microbiology that read a yeast paper and thought like, yeah, we can also do this, and so like, and it could be useful. But yeah, these kind of assays and so like are done and can be very successfully. It's just like the concept I want to tell you. So if you work on something very specific, use all the tools for your specific question. But sometimes if you want to understand, when we say that holistically an organism, it is worth to do what we call a fishing expedition, where you just test all the variables at the same time, and then see what interesting things you find. I never wanted to work on biotin. I never wanted to work on stringent response. When I made the screens, I just added this library and the drug and just said, like, science, show me what is interesting. So, like, and that's why we find all these hits. And so, like, it's just a very different approach than if you start a PhD and someone tells you, like, please describe the active site of this protein, which is very important. And so, like, and in both approaches are important, but sometimes to really discover new things, it's really good to just do it blindly and unbiased and just see what you find. And so, like, and these are these approaches, and my lab is developing the tools for that. And so, like, and if that is also useful for your research, so we collaborate a lot already with the University of Gdansk. So, like, send me your samples, we can test and screen things. And so, like, because we use robotics, so it's actually not so much work. So what I presented is a work like from 2 people, we have 10, so, like, we do much more things. So we really do assay development.
Thank you very much. I will think about this, so thank you.
Yeah.
Wonderful, thank you. Another beautiful example of the opportunities that can arise from our conference, so we're very happy to have this. We also have a question from Adriana Koshiruk. I believe we have time for one more question.
Yes, hello. I have one question. Which bacteria is particularly difficult and dangerous among bioforming bacteria? I mean.
So I think all bacteria are difficult and dangerous. I mean, all pathogens. It really depends on your own, also genetic background. So if you're a cystic fibrosis patient, so you likely die from Pseudomonas, and so, like, like, difficult to work. And I think really challenging for us is we also, and I didn't talk much about that, is so, like, we work with the vaccine strain of tuberculosis, and there the technical challenge is that it takes 4 weeks to grow a plate. Whereas, like, if you work with more standard pathogens like, Staph aureus, Pseudomonas, E. coli, and everything that, and so, like, it just takes a day. And just this difference between 1 week and 4 weeks is just really hard to optimize things, because you try something and then you need 4 weeks until you see the results. So, like, what you can do in 12 days with a fast-growing bacterium, you need a year in a way, like if you would always wait for your result. And so, like, with a slow-growing bacterium. So, like, these are, at least in the lab, very challenging. And the other thing that is challenging if they form a lot of biofilms, because then it gets really sticky. And if you think about that, we actually really use a replicator. It's a stamp, nothing else. And so, like, but if the bacteria are very sticky and gooey, then they transfer different amounts of cells each time. And so, like, then you need more replicates, and you have to try to find condition or even add something in your source plates in the library that they don't form biofilm. And so, like, so that is, like, for us, some bacteria are harder to work with than others. So the easiest, as always, is E. coli, because, like, E. coli was just so evolved to be such a lovely lab pet. And so, like, but as soon as you go to clinical isolates, that it just gets much more harder to do these experiments reproducibly.
Thank you very much for the answer.
Okay, thank you very much for all the questions. And Dr. Banzhaf, we were very happy. Oh, Daria Wada also has a question, I think.
Yes, I would like to ask you a question. How long it took to develop the Herman JPP software, and it will be improved in the future?
So the software, that's I was very fortunate, because all credit goes to Hannah. So, like, I had a really good PhD student that wanted to do some coding work, and she pretty much, like, using tutorials self-taught herself most of the things. So, like, I teamed her up with a real bioinformatician for a bit of mentoring, but if you use the internet right, and so, like, so this is really something something that can be done or achieved to a level like within a year or so. Like, so it probably took a year to write the software, and now we will continuously improve it because it's now the backbone of like a lot of things we do in the lab. And in general, like, I can just hope that whenever you think about do a software, you know, we have I think a Discord server, so just make everything publicly, so like everything for us is open source, and if people struggle and so like, so we still try to help them out and so like, and there is, because it's a GUI, so you can use it without any bioinformatics skills, and so like there's always some limitations because you might want to analyze your data in a certain way we haven't thought about when we made the GUI, because there's just like put your file in, click, and so like then you get everything and you get the figures made. So there can be limitations, but if there would be something, you could contact us and we can see if we still incorporate that. But if you can code Python, then actually if you have the source code, it's very easy to adapt to what you want because we annotated everything quite nicely. At least that's what I have been told, because again, you know, I did some MATLAB and some coding like maybe 7 to 10 years ago, and by now I think I you know, I wouldn't want to write a software anymore. So like now I write grants and papers and give talks and so, but it's definitely possible, and so like with all the tools and so like to, if you think about doing something similar like that, and so like just go for it and so like.
Thank you very much for the answer.
Thank you again. It was a pleasure to have you here, Dr. Banzhaf. Thank you for the captivating lecture. If there are no more questions, I think we are free to proceed with the first session of our oral communications. Therefore, I would like to invite Katarzyna Hoffa from the Poznań University of Technology.
Hello.
Hello.
One moment, please. Could you hear me and see my camera?
Yes, we can see you and we can hear you.
Okay. Hello. One moment. Oh, this is not this presentation. Okay. Could you see my screen? Can you see my screen? Can you hear me?
Yes.
Okay. Can you see my screen?
Also, yes.
Yes, we can.
Okay. So, hello, my name is Katarzyna Hoffa. I would like to present our research work titled Magnetite Polynorepinephrine Cholesterol Oxidase as a hybrid material for biosensor application. I will start with a theoretical introduction. Following that, I will present the aim and scope of the work. In the next section, I will present the obtained results. And finally, I will summarize the entirety in a few sentences. Nanotechnology is a branch of science that studies structures where at least one dimension falls within the range.
Sorry, Katarzyna.
Yeah.
I
Yes.
see only working version of the presentation.
Okay. Now is okay?
No, to same, only the working version, not in present and type.
Okay, maybe now is it okay?
Yes, it's okay.
Okay, thank you so much. Nanotechnology is a branch of science that studies structures where at least one dimension falls within the range of 1 to 100 nanometers. Hybrid nanomaterials among these structures have had an impact on the development of biosensors. Biosensors are analytical detection devices. They operate by reacting a receptor such as an enzyme, antibodies, microorganisms, or nucleic acids with analyte, in this case cholesterol, resulting in the formation of the chemical information. In the next step, this information is transmitted to a transducer, which converts the data into an easily measurable signal, such as thermal, electrical, or optical. And biosensors have a lot of application, for example, in medical diagnostics, including monitoring cholesterol levels. The aim of the research work was to obtain a core-share hybrid nanomaterial in which the metallic core was made of magnetite nanoparticles and the biomimetic coating was polynorepinephrine. Then cholesterol oxidase was immobilized on the prepared matrix. The metallic nanoparticles were synthesized by co-precipitation of iron chlorides under alkaline conditions. The resulting magnetite was coated with catechol compound by polymerizing noradrenaline hydrochloride. Subsequently, the composite was subjected to physical analysis using IFM, FTER, DLS, and zeta potential. The prepared matrix was utilized for immobilizing cholesterol oxidase enzyme. Additionally, the material was deposited on a GC electrode surface and coated with Nafion. And finally, the basic electrochemical analysis was conducted using cyclic voltammetry. Now I will present the obtained results. Zeta potential measurement was used to determine the electrokinetic stability of nanoparticles in a liquid solvent. Negative zeta potential values indicate the prevalence of anionic groups in the structure of tested materials. The material is considered stable if the zeta potential value is equal to or lower than -25 millivolts. The table shows that both magnetite and hybrid material are electrokinetically stable because their value of zeta potential is lower than -25 millivolts. Non-invasive light scattering was used to investigate molecular structure of materials. The measurements obtained for magnetite and hybrid material were 79 nanometers and 168 nanometers respectively. So differences in nanoparticle sizes comes from their ability to aggregate. Additionally, the polydispersity index was used to describe the particle size distribution in the sample. As we can see, the magnetite is monodispersion system. The hybrid material is also monodispersed system, but nanoparticle size are more diverse. The atomic force microscopy was used to describe morphology of the hybrid material. The image from IFM suggests that material consists of a lot of many particles. The image shows brightly yellow spots indicating the presence of layer, most likely polynorepinephrine. The roughness coefficient was also measured. The average value of this coefficient was 9.6. This suggests that material has moderate surface roughness. Fourier transform infrared spectroscopy was also measured. We are used this method to identify the functional groups present in the structure of magnetite and hybrid material. As we can, we observed a characteristic region and peak for OH group. Additionally, in the fingerprint, we observed a special range for magnetite core. The spectra obtained for hybrid material shows us a characteristic region for OH and NH group, which is attributed to catechol groups from, of polynorepinephrines. We also observed peaks corresponding to aromatic rings, and we also observed a special range for magnetite core in fingerprint. Electrochemical analysis was conducted using voltammetry. The analysis was conducted for 2 generations of biosensors. However, today I will focus on the 2nd generation of biosensors, briefly mentioning the 1st generation. The results obtained for the 1st generation of biosensor are very promising. The analysis shows that the process is diffusion controlled. Additionally, the study relieved an increase in reduction peaks with increasing cholesterol concentration. The analysis of the 2nd generation of biosensor system began with testing the impact of different scanning rays on the electrochemical properties of the material, which allowed for the determination of the current versus the square root of a scanning rate curve. A peak anodic and cathodic peak anodic and cathodic show a linear dependence, suggesting a diffusion-controlled electrochemical process. An investigation was also conducted in a solution with varying cholesterol concentration. Here we encountered an issue related to cholesterol dissolution. So we applied a special mixture of Triton X100, isopropanol, and PBS buffer. An increase in anodic peaks was observed with increasing cholesterol concentration. Beyond 50 milligram to deciliter, the voltammetric response begins to stabilize, indicating enzyme saturation. So let's move on to the conclusion. The research conducted and the analysis of the results provide significant insights, allowing the following conclusions to be formed. So physical tests made it possible to learn the structure of individual components and confirm the effective synthesis of magnetite and polynorepinephrine hybrid material. A cyclic voltammetry test confirmed the enzymatic activity of the device, which increased with increasing cholesterol concentration in the solution. And finally, the obtained results confirmed the potential of using the biosensor system to measure cholesterol levels. I would like to express my great thanks to Nano Biosensa team. Thank you for your attention.
Thank you for the presentation. Now, if anybody has any questions, same as before, just raise your hand. Perhaps I will start off with a question. I wanted to ask you what, in your opinion, was the greatest challenge of your research?
I think the most challenging was electrochemical analysis, because we must do a lot of tests, and the most difficult was problem with cholesterol dissolution, I think.
I have one question. Why you used Nathan as matrix to close it nanoparticle as by not other conductivity polymer or carbon paper?
It's a good question, because we check another substances, but Nathan it was the best, and it is ion selective, and prevent to contaminants.
Okay, thank you.
Nie słychać. You have muted mic.
We cannot hear you, Alexander. We cannot hear you.
Your microphone is muted. Try now.
Can you hear me now?
Yes.
Yeah.
Oh, okay, sorry. It appears that my Teams has frozen and I can't do anything. Okay, if you can hear me, then thank you for your presentation. I would like to invite the next speaker, who is Maria Kuznowicz, and she's also from the Poznań University of Technology. Are you with us, Maria?
Yes, I am.
Hello.
Okay, hello. Thank you for the nice introduction.
No problem.
Okay, is the presentation visible?
Yes, we can see.
Yes. Okay, so thank you. So my name is Maria Kuznowicz, and ladies and gentlemen, I'm really pleased to present you my research titled Miniaturization of a Glucose Biosensor based on polycathodic acid hybrid material. And today's presentation will be divided into 4 main parts. In the first, I will present you and give you the key important information about the biosensor, but also about the problem with today's glucometers. Then I will go to the aim and the scope of the part of this research and present the most important, of course, experimental part. And finally, I will conclude this and compare and show the sum up of this research. So my friend in this previous presentation present you a biosensor, which is a type of analytical devices which convert the information from biological domina to a measurable parameter. And mostly they are the most important part of the biosensor, and which I also focus, is the receptor layer. And because we can use a lot of different materials like enzymes, carbon nanomaterials, metal oxides, polymers, or metal nanoparticles, but what is important that the choice of this material can really affect on the final properties of the sensor. I mean, mostly of the selectivity, because we would like to measure just only the one component which is for us important, their sensitivity, because it's also one of the key parameters, the stability, I mean mostly in different temperature or pH. Also response time, because of course right now we would like to have really quick parameters, and also sensor lifetime, because one of the problem of today's glucometers on the market is their lifetime. And I would like to tell you more about that in the next slides. So maybe you have a question why we still work on the glucometers when we have a lot of them on the market. Is that true that they have a lot of different limits and errors, mostly because of the aging of the nano platform, but also this biological element, the manufacturing process and appropriate storage, problem with the use coding of these devices, and also really one affecting parameter, which is a temperature. And because of that, during the studies, I create three research hypotheses. First, that the hybrid nanomaterials can significantly affect on the sensitivity of the sensor. Also, that the use of biopolymers coating can increase the efficiency of the immobilization process. And also that just the immobilization of the enzymes on the surface of hybrid material can really improve the electrochemical selectivity and mostly time stability of the sensor. So the main aim of the work was creating a hybrid material, and after that the use of them in the biosensor application to obtain device which can be really more selective, more time stable, more sensitive, also which can be of course cheaper, with this effect of miniaturization, to finally use them in the tests on model and real solution like blood or the human serum. So to go to the experimental part, here you can observe the general idea of the work. And the first step, the magnetite nanoparticles was obtained with, after that, which was covered with polycaffeic acid film. And this platform was used for immobilization of the enzyme, adsorption, immobilization of glucose oxidase. And after that was used for the modification of electrode. In the case was used screen printed electrode, which allow us for the miniaturization of the system. Maybe to compare classical electrochemical cell use like 5-10 milliliters of analyte, which is needed for the nice tests and to obtain the real data. But in this case, we use like 40 microliter of analyte, like human blood or human serum, and to get our results. So the miniaturization effect was here presented. But like I said, the use of this receptor layer, I mean the hybrid material, is one of the most challenges right now in our research. And here you can observe the use magnetite polycaffeic acid hybrid material. Magnetite nanoparticles with the size of 8 to 12 nanometers were covered with polycaffeic film in the size from 2 to 3 nanometers. After that was used, as I mentioned, for the glucose oxidase immobilization. And here on atomic force microscopy image, you can observe this matrix before and after immobilization of glucose oxidase. And here it's important that we observed a zeta, the increase in the zeta axis parameters, which show us that the efficiency of this process. Also, we check it with Bradford analysis, and we check the immobilization, different time of immobilization of this enzyme, and we compare the just native magnetite nanoparticles with our hybrid material. And what we can observe here is the fact that the best results we observed after 24 hours of enzyme immobilization, and that the use of polycaffeic acid as a biofilm was allow us to improve the immobilization process when we compare them with just a magnetite. But of course, one of the most important part was the electrochemical test to check how the system works, if it works properly, and how. And here we can observe that the system works with different glucose concentration from 0.505 millimole of glucose to 25 millimole of glucose. And what is important here, that this allowed us for measure the glucose concentration in the with the guidelines of World Health Organization, which present the glucose concentration, which have to be measured to present and to create new glucometers. And with our system, we can measure our glucose concentration from hypoglycemia when we observe this lower sugar level in our blood, to even hyperglycemia when this sugar level is too high. We also check if our system is selective with this use of hybrid material. And you can observe here that we check different sugar and also the popular compounds like uric acid, ascorbic acid, which we can observe in our blood. And what is important here, but the relative response which is presented here was the higher from the glucose, and the adding of different interference didn't show a good response. And after that, we can say that our system is really selective. But I think one of the challenge, as I mentioned, is time stability of the system. Maybe here to compare the commercial glucometers works properly and works usually to 3 months after the use. So is, I think, one of the challenge in general when we talk about the glucometers. Our system works properly even after 10 months from the creating, with still the relative response, which we can use them with this World Organization guidelines, and it's still like more than 3 times better and longer than the commercial glucometers. And after this basic test, we check how this system works in real samples. So we measure glucose concentration in two different samples, I mean in the human serum and human blood, with different glucose concentration from hypoglycemia to hyperglycemia. And what you can observe here that the recovery of the presented biosensor was above 97% for all the samples. So these results, of course, are not like 100, but we are still in progress and still work on our devices. And in conclusion, the presented biosensor was made based on the hybrid material magnetite polycaffeic acid and used for the glucose oxidase immobilization, and work properly even after 10 months from the construction, and was characterized with great selectivity and the linear range corresponding to the guidelines of the World Health Organization. In this moment, I also would like to thank you to Professor Topil-Jesionowski and Dr. Artur Jędrzak, and also to the Poznań University for the research grant. And of course, I would like to thank you for your attention.
Thank you for your presentation. I would now like to open the floor to any questions. Perhaps I would like to ask a question, specifically, why did choose exactly this area of study to go into?
I think because of the problem of the diabetic, which is really big problem today, and also with the problem which we observed, and is a lot of publication which we read, but the problem with glucometers right now. So when we observed a really big problem like diabetics right now, and also, but we know that the commercial glucometers not work properly, then we see that something has to be changed, and because of that we work on the glucometers.
We don't hear you, Alexandra. Okay, so I have a question. Why did you choose this polycaffeic acid for the biosensor? Did you think about the other polymers?
We work with mostly with the catechol polymers because of the electroactive properties, like from the electrochemical application. We check also like polydopamine and polynorepinephrine in different studies also, but also the polycaffeic acid, maybe I didn't show here, allowed us for the lower potential. I mean like 0.1 volt, like with different polymers and with the mediator, we need like 0.2, 0.3 volts. So we have lower potential, and this allowed us for the more selectivity, and because of that. And we mostly focus about with the biopolymers because of the properties.
Okay, thank you very much.
Thank you for the question. Can you hear me now?
Yes.
Okay, perfect. I do believe that there is a question that was written on the chat just now.
Okay.
What is the main thing that you think is better about this technique than a test available widely now in the market?
I think the one thing is the time stability. I think is the most advantage when we compare it with commercial one right now. The technique is the same because we also use the electrochemical sensor, which is on the market. I think the use of the material right now, that we use biopolymers, is better in this case because we focus on the immobilization process and with the time stability. I think this is the main advantage when we compare it to the commercial one.
I see. That question was from Lisa Amalia, if I'm correct. So thank you for that. And I don't see if there are any more questions. If not, then thank you for your presentation, and we will move on with our next speaker.
Thank you.
Thank you. It's Kornelia Hyjek from the Krakow University of Technology.
Hello, I have a problem with my camera on a computer. So I show my screen by a computer, but turn on my camera on the app with the telephone, if there is no problem.
That's not a problem at all.
Okay. Okay. So could you see me?
I'm afraid somebody else will have to confirm that. Yeah, we can see you.
Okay, okay. So I show. Okay, sorry for that. Okay. So I would like to say something about the in vivo and in vitro studies of effective methadone absorption over zircon-based metal organic frameworks corroborated by DFT plus D modeling. And firstly, I would like to say about the problem of drug addiction and answer the question why the problem is so crucial. Maybe you don't know, but the number of people addicts of drugs increase every year, and this problem occurs not only to adolescent and young people, but also in the adult. The range of novelly discovered compound with psychoactive effects is growing, and the main problem is to make them illegal, because identify these substances is really problematic. One of this new group of psychoactive substances called synthetic catechins become more popular in recent years, and the representative of this group is methadone. And methadone is easy availability. The price of methadone is also very low, and the methadone have an amphetamine-like effect, so it's promoting to popularization of these substances. And we have two way to fight an overdose. Modern medicine recommends to use to do the gastric and intentional lavage, and is suggested in overdose situation. But also we can administrate the substances with antagonist effect to the drugs. For example, if you methadone poisoning, you should administrate the propranolol, because the propranolol substances have an antagonist effect to methadone, and help to eliminate the effect of overdose. But in the medical market, we don't have any medical adsorbent which can be suitable and safety for overdose situation. If we think about medical absorbents, we think about active crackle, but the use of active crackle during overdose could be dangerous because of trachycardia or dehydration as a result of rapid removal drugs from the body. So we can conclude that the pharmaceutical market lacks suitable adsorbents that can be effectively and safety for human during overdose. So we propose to use the metal organic frameworks as a perspective approach to solve the problem. Especially we use MOFs like medical adsorbents, and is totally new concept. The metal organic frameworks is a new group of mesoporous materials with three-dimensional structure, consists of the metallic part, like inorganic part, and organic part, carboxylic acid or another organic acid. And thanks to this, thanks to the build and undefined structure, the MOFs have unique characteristics, such as high thermal and chemical resistance, large specific surface area, and a high bioavailability and low toxicity. By bioavailability and toxicity is depend to the metal ion in this structure. And MOFs become more and more popular in recent years, and as a result have a lot of application, for example catalysts, gas adsorbents, even hydrogen storage, which is really popular, for example in a transport, and drug carriers. But use MOFs like medical adsorbent is a new approach. And we selected and study zircon-based MOFs called UO66 with hydrochloric acid, and the NU100. And the common features was the presence of zircon as a metallic part. This MOF consists of different ligands, and thanks to the different ligands, this results in differences in crystallinity, with structure, porosity, and as a result, properties. We synthesize MOFs by solvothermal methods, and then the materials was activated under the vacuum at high pressure and study physicochemical. Thanks to powder X-ray diffraction, we can confirm the correctness of the study and the crystallinity of the sample. And by the nitrogen adsorption isotherms and DFT pore size, we know the nature of the materials, especially the specific surface area and the pore size. The most important in our study was the transmission infrared spectroscopy. We did 3 kinds of spectra: the pristine MOFs, the mephedron substances, and the spectra of composites. The composites, I mean, it's MOFs after the sorption process. So the MOFs with mephedron inside. And thanks to this study, we confirmed that the mephedron is effectively loading to the MOF structure, and it is the evidence to mephedron in MOFs. An additional study was the scanning microscope electroscopy, and this study allowed us to observe the structure after the absorption process, the morphology and the topology of the prepared sample after the adsorption. And absorption of the mephedron was conducted on 24-hour, and we used the medium, distilled water as a medium. But for the best result, which was obtained by NU100, we do the additional study. We use a simulated human body fluid as a medium, and we can observe that the amount of mephedron, which is absorbed by NU100, is the bigger. We also calculate the amount of the maximal amount of mephedron, which can be absorbed by the materials. And this study emphasized that the beneficial effects of modulated metal organic frameworks. So if we add the hydrochloric acid or another modulator, it causes easier access to the MOF channels, and as a result, the adsorption will be better. And the best results was obtained for NU100, and the adsorption profile and the kinetics is really gentle, but also fast and effectively. And we observed that if the metal organic frameworks have a contact with the simulated body fluid, and the contact with the ions in this solution, the hydroxyapatite layer is formation on the MOF structure, and thanks to it, the adsorption is more effectively. And is really crucial and untypical dependent. And if we want to use MOFs as a medical adsorbent, the biological study is necessary, so we did it. And the biological study confirmed that the MOFs, their MOFs no toxicity effects, and the absorption the absorption is effectively. We also check the parameters like heart rate and locomotory, and it is also evidence to no toxicity effects of MOFs and the composites. And we conduct the color preference test. This is the kind of test which can confirm us to the effective, to the addictive potential of the substances, and thanks to this test we confirmed that the mephedrone is addictive, of course, but the MOFs and the composites don't have addictive effects. So the metallic organic frameworks can be used as a modern adsorbent to remove psychoactive substances from the body during overdose, and the proposed therapy is safe and highly effective. And also the ion contained in the human body increase adsorption efficiency. And this is also really really good for us, and the really perspective for the future study. And the results presented during my speech were published in the article, and the title of the article you can see in the page. And we also studied the substances with antagonist effect to mephedrone, and it is the propranolol, and use metal organic frameworks to deliver, to administrate propranolol during overdose, and to fight with withdrawal effects of mephedrone. And the work was supported by National Science Center under the research project. And that's all. Thanks for your attention, and encourage to have a question.
Thank you for your presentation. Like Ms. Hyjek already said, if anybody has any questions. Oh, Mateusz Baruch.
What is the cost of the synthesis in 1000 and UIO66? This is cost effective?
The new 1000 is really the cost is really high because of the ligand. The ligand is the acid based on a appearance structure, and is really costly, and it's really hard to synthesize this because you don't have to buy this ligand. You must to synthesis by your own. So it is the long and cost, but the UIO66 is the synthesis is really really simple and rather cheap, because the ligand is the terephthalic acid, and the zircon fluoride is the metal part, so it's the rather cheap substances.
And one more question, between MOF synthesis and activation in vacuum dryer, yes, how do you clean MOF?
We washed MOF with methanol and dimethyl formid, DMF.
Okay, thank you.
Thank you.
Does anybody else have any questions? Anna Axman?
Yes. I'm curious if you tried effectiveness of this material to remove from the organism also other drugs.
Yes, we conduct study to remove the cocaine, the amphetamine, methamphetamine, and mephedrone, and also MDMA. And we use also zircon-based MOF, zircon-based metal organic frameworks, and we also confirm the effectively of the process, and also biological safety for human body. And we also conduct the study to administrate the antidote, like a propranolol, like a naloxone during opioid overdose, and and that's all.
So this material seems to be quite to have a broad range of activities. So not only for the strictly one drug, but also for similar drugs. Okay, thank you.
Yes. For similar drugs and lots of substances. For example, we also use MOFs like anti-cancer drug delivery system and anti-cancer therapy using a novel approach with inhalers to lung cancer. So it's also another application.
Very interesting research. Thank you very much.
Thank you.
Thank you very much for that presentation. We will move on to our next speaker now, the last speaker of the first session, which is Maja Radecka from the University of Cambridge.
Hello.
Hello.
Can you hear me all right?
Yes, I can hear you.
Mm-hmm. Can you see the presentation screen okay?
I can't see it yet, but maybe somebody else could confirm if they can see it.
Okay. So today I'll be bringing a little bit of physics to the discussion, and I'll show you the results of my master's project, which concerns atmospheric chemistry in exoplanets. So, the title of my project is Nitrogen Content of Hycean Worlds, so I should start with explaining what are Hycean worlds. It is a very recent concept developed in roughly 2021 by Niku Madhusuhan, and Hycean basically means hydrogen and ocean. It describes a planet that is roughly the size of this planet I'm concerned with, K218b. So less than 10 Earth mass and roughly between 2 and 3 Earth radii. And judging by mean density, we could have 3 options that this planet could have. So it could be a rocky world with very thick hydrogen envelope. It could be an ocean world with slightly less big hydrogen envelope, hence Hycean world. Or it could be a mini Neptune. So for this particular planet, the most plausible options are Hycean world or mini Neptune. So these two I'm going to focus on while doing my measurements. And why K218b? So this planet is very interesting for us because it receives almost the exact same amount of solar radiation from its star as Earth. If you look at the diagram, it would be very close to terrestrial habitable zone. Here, this yellow dot, that would be where K218b is. And why do we look at Hycean worlds anyway? It's because of a very thick hydrogen envelope. So hydrogen is a very good greenhouse gas, and it can retain heat much better than other greenhouse gases on Earth. So what this means is that the planet can receive less radiation and still contain and still be within the temperature range for liquid water to be present on the surface. So if the planet is far further away, so called Hycean, if it received some certain amount of heat initially and then migrated further into the outer edge of the solar system, it could still be habitable, as well as if it's too close, so it receives too much radiation, but on the night side of the planet without any light from the star, life could technically exist. So once we talk about the possibility of liquid water on the surface, we could consider whether clouds could form in the atmosphere. And in order to do that, we need to compare partial pressure of water with saturation vapor pressure. So as long as the partial pressure of water in this atmosphere is larger than saturation pressure, water could condense and we could have clouds. And in order to check that, we can use Antoine equation and to plot condensation curve for water or other molecular species. And that's what I've done here. On this graph, you can see temperature profile for this planet, which is given from Nikus Madhusuhan data. So dotted is the function of pressure depending on temperature. And I'm also showing water and ammonia, partial pressures and condensation curves. As you can see, condensation curve intersects partial pressure of water, hence clouds could be possible. It's not the case for ammonia because K8b is too hot for ammonia. So ammonia clouds wouldn't form on its surface. However, for example, in Jupiter, which is much colder, we would have ammonia clouds. Yes, so there is possibility of liquid water if we assume that this temperature pressure profile is correct. And here you can see the measurements from James Webb Space Telescope. This is very recent data from 2023. And you can see transmission spectrum and loads of absorption bands of methane, some CO2, and DMS. In previous measurements made with Hubble, this planet showed some water spectrum. However, I'm not focusing on water and dimethyl sulfide because both of these, the presence of both of these molecules is inconclusive. And so I'm only focusing on molecular hydrogen, methane, and carbon dioxide. And to put these numbers into a bit more digestible concept, they would translate roughly to around 1% and 2% amounts in the atmosphere. So what exactly am I doing? How am I simulating the atmosphere? I'm using 1D photochemistry and diffusion code developed by my supervisor, Dr. Paul Rimmer. And what it does, it takes a pulsar of air at the bottom of the atmosphere and then moves it upwards and then back down. And at each height step, it checks the chemical network of over 6,000 reactions put into that code. And for each species, it checks how the number density changes with time, depending on reactions that produce the species, depending on loss reactions, and also the flux of these particles. The change in flux is calculated for both molecular diffusion and also just vertical convective mixing, simulated as eddy diffusion. So from this code, we could produce height profiles of different molecules. And here is how this height profile would look like. So this is my reference sample without any nitrogen or any extra inputs. My only inputs are the ones from the James Webb measurements. So here we can see that some water, carbon monoxide, and organic carbon hazes are produced purely through photochemistry and from these inputs in this atmosphere. One thing that is common in all my measurements and is a bit concerning is that we have quite a lot of CO production here, which should be in principle detectable, but we did not see it. So now to move on to samples with nitrogen. This is my most interesting sample because it produces the most of nitrogen-bearing compounds. And it's sits at extra nitrogen added at 1%. So we have still H2, CH4CO, and also nitrogen added here at 1%. We have similar production of organic hazes of water and CO, but we also have some photochemistry happening in the upper parts of the atmosphere, which produces ammonia, hydrogen cyanide, and cyanoacetylene. However, looking at this orange rectangle, this is roughly what James Webb could see in the atmosphere. So it cannot go below 10 to the -5 mixing ratio, because then the spectrum is not strong enough. We don't have enough molecules to actually produce a good signal. And it only probes this part of the atmosphere between 10 to the -2 and 10 to the -5 bar. This is because it has to be above clouds, but also the higher we go, the less molecules we have, so the less strong signal. Another thing we need to test is whether K2-18b is a mini Neptune. So now I'm checking mini Neptune versus high sea in world approximation. In order to do that, I've put in 100 times solar metallicity. Metallicity is the amount of other elements compared to hydrogen. So in this sample, we have 100 times more other elements compared to hydrogen than in our sun. Dotted curves mean chemical equilibrium, and solid and dashed lines for just photochemistry and all the diffusion happening. So as you can see, it predicts well for methane because it gives roughly the same order of magnitude. However, this result gives us way more water and way more ammonia than we would expect to see from the measurements. So this hypothesis that K2-18b is a mini Neptune is not so well fit. However, it shows CH4 well. And to combine all of my nitrogen measurements, you can see here that all of these curves peak around 1%. And here on the left axis, this KZZ means the amount of eddy diffusion. And as you can see, if I change this amount, it doesn't have that much effect on the values here. The curves are fairly similar, and it is common that for all of them, 1% produces the highest amounts of other nitrogen-bearing compounds. So to conclude, this is a very growing field now due to recent development of James Webb, and there are way more measurements to come in the future. So I'm really looking forward to having more data on it. It is also very, so the field is still very inconclusive. For example, we don't have full stellar spectrum. We only probe the star in the wavelength range of James Webb. So if we have the full stellar spectrum, we could have completely different result. If we use 3D instead of 1D molecular diffusion code, we would also probably have a different result. So it is all very speculative. However, this simple approximation can show us roughly the amounts of molecules we would expect to find. And so far we see that N2 would not produce enough nitrogen-bearing compounds to be detected. However, if we do find them, it means that there must be additional source of them that we did not consider. Could it be life? Could it be just some geothermal process? We don't know. We just have to wait for more measurements and see what comes. Thank you.
Thank you very much for that interesting presentation. If anybody has any questions. I think that there is a question on the chat. No, that's a previous message.
Anna?
Sorry?
Yeah, I have to say that it was very hard to follow for me because I'm not very good at physics. So I have maybe very basic question. What could be the practical application of this research? Because, you know, I understand that it is very interesting, but maybe we can use it for some purposes in the future.
So this research, it is true that it does not have many practical applications. However, developing these atmospheric models for exoplanets could also help us understand what is happening on Earth and how Earth's atmosphere developed the way it is. But this is like a very long-term goal. It wouldn't happen for a couple of papers. We would need to work on way more planets and way more different codes to actually understand what's going on. But kind of the main point of the research is to find if there is life in the universe, if there's some more, like an Earth twin maybe somewhere. Yeah, so it's obviously very optimistic to find it, but yeah.
Thank you very much. It's still very interesting, and because I was wondering if your research can also be used to predict if there are some globes or something like this in the space that can be potentially similar to Earth or something like this, or?
In principle, yes. In practice, it's much harder because Earth is a much smaller planet with atmosphere not being so thick compared to its size. And when we do transmission spectroscopy, the ratio of planet size to the star size matters. So if the star is small and planet is large, then we can observe this very large dip in the stellar spectrum, and we can have clearer measurements. However, if we look at Earth-type planets and Sun-type stars, this is the difference in the sizes is so big that the spectrum is still visible, but not as good as for larger planets, hence like K2 and B.
This is very interesting. Thank you very much.
Thank you, Professor Axman. Are there any other questions? I'm not entirely sure if anybody's raising their hand.
I suppose somebody is waiting for.
Yes, I'm afraid my Teams has frozen, so I can't see, but I believe Martina has raised her hand.
Yes, yes. Hello, do you hear me?
Yes.
Yes.
It is very, very interesting subject, I think. And I have a question because some time ago there was an open project in which you could search for exoplanet using website, and maybe you have some information about this. Maybe is there any project currently open to search for exoplanets for a normal guy?
So I don't know the exact name of the website, but loads of data is accessible to everyone. So some of new James Webb data is restricted only to scientists who ask for it in the recent grant, but a lot of exoplanet detection data is available. I think just for exoplanets.org or something like that.
Okay.
Yes, so it's definitely worth searching on web. And I encourage everyone.
Okay, thank you so much.
Okay, I believe this concludes then our first session of oral presentations or oral communications. I would now like to, before we proceed to the break, I would like to invite everybody to take sort of like a collective picture in the form of a screenshot. So if I could ask everybody to just turn on their cameras so that my group could take the picture. We'll just wait for a couple more to turn on their cameras, and then my team will take the screenshots. And once we have confirmation that we have it, then we'll proceed with the break.
Okay, thank you.
Thank you very much. So now we're going to have a break. We'll be back here at 10:30 UK time, which is 11:30 Polish time. And thank you to everybody who participated in this session. And we'll see you in half an hour.
Okay, so hello everyone. It's 10:30 in the UK, so we can start. My name is Wojciech, and I will be leading the second oral session of our conference. And the second session contains video recordings, and the first one is prepared by Agnieszka Siwiak from Adam Mickiewicz University, titled Challenges in the Synthesis of Non-Centrosymmetric Alane MOFs. So please start the recording.
Hi, my name is Agnieszka and I work as a synthetic chemist. I believe that challenges are an unavoidable part of every synthetic work, because at first we set ourselves goals, such as synthesis of novel, smart material with interesting for us properties. We start to work, we get our first results, and then we confront the reality. The work appears to be not as simple as we thought in the beginning. But let's start with what my goal is and why. So my goal materials are included in a wide group of materials called metal organic frameworks. As the name suggests, they are built from organic part, called linker, and inorganic part, which is called the node. As you can see, linker has at least 2 atoms which can coordinate with the node. Really often it's nitrogen or oxygen. On the other hand, nodes are the most often metal ions. What will be important to notice further, if the node is lanthanide ion, for example here, the obtained MOF is called Ln MOF. MOFs are quite popular and fast-growing class of materials because they are porous, crystalline, and have large surface. Thanks to the rational choice of components and appropriate functionalization, they can find application in a variety of areas. Some of these applications are rather obvious. For example, they are porous, so through guest-host interactions, they can store gases. However, what is more interesting to me is their potential as sensors. In the literature, you can find diverse types of sensors, although I'm focused on temperature sensors. Because just think for a moment, when was the last time you took the temperature? Maybe you felt sick, maybe you wanted to know how to get dressed, or maybe you just wanted to conduct reaction in specific conditions. However, we must say that the temperature measurement is really important in our lives. Although all these measurements are made in macro scale and demand physical contact with the sample. To overcome these limitations, promising for example in medicine, we decided to work with Ln MOFs, which thanks to the intercorporation of lanthanide ions, exhibit luminescence properties. And when we take a closer look at the luminescence spectrum, we can observe how some parameters vary depending on the temperature. In the conclusion, when we know the spectroscopic properties of our materials, we can excite the material from the distance and then determine the temperature. However, my workmates decided to try something new in the field of luminescence thermometry. They combine nonlinear phenomenon guaranteed by non-centrosymmetricity with the mentioned luminescent properties. As the linker, they chose formic acid, which is really simple and really cheap. But what's the most important, as I mentioned before, MOFs are crystalline materials, and depending on conditions of synthesis, various phase groups of MOF containing formate may be created. As the other component, erbium doped with ytterbium was chosen. First of all, erbium have thermal coupled levels. Moreover, the emission is in the range of biological window, so it increases material potential in biological applications. And finally, the doping with ytterbium improves optical properties of our material thanks to resonance energy transfer. And last but not least, space group, which is crucial for this material. So thanks to it, thanks to the lack of the center of symmetry, it exhibits second harmonic generation phenomenon, in which 2 photons with lower frequency are converted into 1 photon with doubled frequency. Because its signal is also dependent on the temperature, we can take the ratio of luminescence intensity and second harmonic generation intensity to get the linear relationship. And this relationship enables us to accurate temperature measurement in indirect way. Our next step was to implement the same idea on a different linker. Trimesic acid was rather logical choice because it's really popular and classy linker. Even the first reported ever MOF contained it. Moreover, it's rather cheap and for sure bigger than formic acid, which is really promising because it gives us space for functionalization of MOF, which can potentially improve spectroscopic properties of our material. So I carried out my first test hydrothermal reaction in Teflon flask. At first, I decided to work on a system containing only erbium ions because they are crucial for our optical properties. And I decided also to work with the mixture known from the literature, so DMF and water. After some days, I drained the sample and I took it to the PXRD analysis, and as you can see on the diffractogram, it didn't work. The material which I obtained was centrosymmetric, so I carried out some more experiments and finally I found out where was the mistake. The main problem was with the conditions of my reaction, so I repeated it, but now with lower temperature and shorter time. So to be totally honest, I just did it the same as in the paper. And as you can see here, after the PXRD analysis, everything looks fine. So all the peaks are in the same positions. So we've got non-central symmetric material. Being really optimistic, feeling that work is finally going forward, I gave my precrystalline pink material to Przemek, who specialized in testing of optical properties. After some time, he made preliminary tests for my samples and told me about the results. The results were just really bad for me. He said me that he didn't observe second harmonic generation phenomenon, and that, to be honest, he thinks that these samples are not suitable for optical applications. And to be really honest, these optical applications were for me the most interesting part of my project, so I was really disappointed. So a new challenge has arisen to me. What may be the cause of the problem? Because we have to overcome it now. First thing, which was also suggested by Przemek, was the laser power. Because, yeah, its power is one of the factors which have influence on second harmonic generation intensity. We have greater laser power, we have also greater second harmonic generation intensity. Sounds logical. However, I still have in my mind that if I want my materials to be potentially applicable, they cannot demand conditions which are difficult to meet even in laboratory to measure in this indirect way the temperature. Next idea was that maybe there's something wrong with crystal structure. Of course, I've got PXRD results and I analyzed them. They seem to be fine. Although single crystal XRD results are more clear, especially when we do something for the first time. So I talked to crystallographer if we can measure my material. I showed it to him and he said that it is crystalline, although we cannot measure it because it looks like the crystals are too small. And that was, I believe, crucial moment for me because that's the moment when the idea has arisen for me that maybe the problem was just with the size of the crystallite which I obtained. And it seems to be a good clue from two perspectives. So first of all, if I obtain bigger crystals, I can take them to single crystal XRD analysis, and the results can show me the full structure of my material and understand it better. And moreover, second harmonic generation is not only dependent on the laser power, but it's also dependent on the particle size. The correlation is maybe not so obvious, and it's different for phase matchable and non-phase matchable cases, although it's not a problem to check how it's gonna work with different size of crystals. And even if the crystal size is not a solution for my problem in this project, I still think it's really worth a try, because when you look closer to the current literature about MOFs, you can find voices that the synthesis of large MOFs is underexplored, especially in the comparison to nano-sized MOFs. And actually, when you look closer at it, the synthesis of bigger crystals is an interesting challenge. In the paper with such poetic title as Metal-Organic Frameworks, in Pursuit of Size, the Development of Macroscopic Single Crystals, some factors which may have influence on the size of crystals were discussed, such as reagent concentration, pH control, reaction temperature, modulation, reaction time, or solvent choice. Although, as you can see, there are a lot of them, and we have lots of possibilities. So the rational synthesis requires better understanding of MOF crystallization, which kinetics is a complex process. So if it's complex, it's also, I believe, interesting. And I really hope that my further work on this project gives me possibility to understand better this complex process of crystallization. I hope that the new synthesis with different conditions, new methods, will give me some more details about how it really works. And to sum it up, although synthetic work makes us overcome challenges, which sometimes may be really hard for us in the beginning, I believe it gives us also opportunity to explore the details which we haven't considered as such important as they really are. So thank you a lot for your attention, and if you have any questions, I will try my best to answer them.
Okay, thank you for your presentation. As I can see, you're already turned on your camera, so perfect. I see that Dominika wants to ask one question, so please, Dominika.
Hello, I have one question. Do you have an idea for the future research?
So if I understand correctly your question, you asked if I have ideas for my future research. So
Yes.
now I'm focused on the synthesis of my LNMOS, but with different parameters, such as it was mentioned in the end of the presentation. So now I'm focused on preparing MOS with, for example, different temperature, different period of time, or different concentration of the substrates. And then I want to measure them to have a connection between the parameters and the size of MOS.
Okay, thank you.
Thank you very much. I see that Kornelia wants to ask a question either.
Hello, I don't have a question, but I can give you some advice because I also synthesize metal organic frameworks, and you can try use glass bottle to synthesize, because we sometimes synthesize with on a glass bottle, not autoclave, and I observed that the structure sometimes is different, the crystallinity, the porosity, and I think you can try it.
Thank you. I would gladly try it because I haven't used it before in my lab.
Okay, thanks.
I don't see any more questions from the audience, but I have one that's basic, really. What properties makes a metal a good node in general? Is there only metal nodes, or there's from the other materials nodes?
So I believe that it's important what you want to have this MOF for. So in my applications, for me, it's important to have lanthanide ions because they're 3+, so it's they can coordinate with linkers. And also, which is important for me, is their spectroscopic properties. So they can exhibit luminescence, and this is crucial for me.
Okay, got it. Okay, thank you very much. So, is there anything? Thank you, Agnieszka. It was really interesting, and so we can go further for the another presentation, for another recording. Now it's time for Angelika Nowak from Wrocław University of Science and Technology with a presentation titled Microplasma in Analytical Chemistry, Miniature Sources for Elemental Analysis. So please start the recording. Okay, we have some technical troubles. We'll manage with that right away, so please stay in touch.
Microplasma in analytical chemistry. Hello everyone, my name is Angelika Nowak and I am a PhD student in chemical sciences at Wrocław University of Science and Technology. Today, I would like to provide you some information about microplasma in analytical chemistry. During presentation, I will focus on miniature devices based on microplasma for elemental analysis. My talk is divided into a few parts. At the beginning, I would like to explain you the difference between plasma and microplasma. Secondly, we will move to application of microplasma and devices based on microplasma. I will mention some details connected with 3D printing in the context of constructing miniaturized systems. At the end, I'm going to summarize all presentation. Plasma is created when enough energy in any form is given to a gaseous medium. One way to define plasma is an ionized gas that is mostly made up of atoms, free electrons, photons, and ions. Plasma can be categorized as thermal and non-thermal plasma. High temperatures and high pressure along with heavy electrons are necessary for the production of thermal plasma. On the other hand, low energy is needed to form a cold plasma, also known as low temperature plasma, at a temperature range of 30 to 60 degrees Celsius in any atmosphere or partial vacuum. Cold plasma is the term used to describe the ionized gas created by relatively low energy and electronic density. Now let's move to explanation of microplasma. Microplasmas are low temperature plasma that feature microscale dimensions and a unique high energy density and a non-equilibrium reactive environment, which makes them promising for the fabrication of devices for diverse applications. Microplasmas combined with gaseous or liquid media at low temperatures and atmospheric pressure open new ways to form advanced functional materials and devices. Right figure shows 3 microplasma-related focus areas. Fundamental research in nanoscale synthesis leading to new materials and functional properties. Integration of the plasmas and plasma-processed materials into device fabrication processes, and microplasma applications in commercial devices and systems. Microplasma applications in the development of materials and devices represent a relatively new field with a clear trend of continuously increasing numbers of publications, intellectual property disclosures, and reported commercial applications. One of the reasons for this trend is the many possibilities to use such plasmas in synergy with gases, liquid, and mixed phase media. Representative applications of microplasmas include light sources for multipurpose as light sources for photochemical materials processing and spectroscopic materials analysis, surface disinfection, water purification, and active electromagnetic devices based on artificial microplasma, optical materials, and other devices and systems, including the plasma transistor. The development of highly sensitive miniature systems for the elemental analysis of small samples is required in medical and environmental fields. For example, such systems are expected to be applied to elemental analysis. Recently, there has been increasing interest in the use of micro-total analysis systems as miniaturized analytical systems for the analysis of small samples amounts. The use of such microtest device enables the miniaturization of analytical systems, the reduction of the amount of samples and chemicals necessary for analysis, and a decrease in the analysis time. System like this can replace inductively coupled plasma atomic emission spectrometry that has been widely used for the high sensitivity analysis of trace elements. In this method, a plasma with a volume of approximately 10 cubic centimeters excites and ionizes the samples. For comparison, a microplasma has characteristic length of the plasma is on the order of micrometers. On this slide, you can see microplasma source for microtas incorporated microchip with a microchannel. In this case, dielectric barrier discharge was used to generate a stable plasma at atmospheric pressure. This method is used because the electric barrier discharge allows to suppress the temperature rise of the electrodes and operate it for a long time because of suppression of the current and reduction of the electrode wear via the electric interposition between the electrodes. It is expected that the analytical system can be miniaturized because helium plasma is generated in the microchannel contained in the microchip. As a result, the microchip with a microchannel with the diameter and length of 500 micrometers and 18 millimeters respectively was fabricated. In addition, a pair of 3 millimeters long electrodes was placed on the microchip to generate the plasma in the microchannel. In this study, to prevent damage of the microchannel, a low discharge power and frequency were used. The microchip was fabricated via 3D molding process based on microstereolithography and copper electrodes were placed on the microchannel. The excitation temperature and the electron density of the plasma were spectroscopically measured to evaluate the basic performance of the fabricated system. To prepare the analyte sample was injected and vaporized in a vessel filled with helium. Next, the gaseous sample was filled into a microsyringe and was mixed with plasma gas using a syringe pump. The emission spectra were measured using a multi-channel spectrometer. Figure 5 shows emission spectra of 40 ppm gaseous chlorine. As you can see, it was successful detection. We can see the chlorine atomic line at about 894 nanometers. The bromine and iodine atomic lines were also successfully detected, what shows figure 6. Emissions from gaseous chlorine, bromine, and iodine were analyzed using the plasma source, and it was found that detection limits for these analytes were 0.22, 0.18, and 0.14. The obtained detection limits are smaller than the approximately 1 ppm detection limit of gas chromatography thermal conductivity detector. A comparison of the helium ACP to the developed plasma source shows that the excitation temperature of the developed plasma source was lower by 1000 kelvins. The calibration curves for gaseous chlorine, bromine, and iodine were also plotted, obtaining coloration coefficients of 0.975, 0.955, and 0.986, and showing good linearity for the developed plasma source. In that way, high sensitivity analysis for small sample amounts was possible by use of microplasma source. 3D printing is devoted to construction of an eventual object by sequential deposition of chemical material. Organic materials are the most common, but there are also available materials like ceramics or metals. Fused deposition modeling is the most common type of 3D printing, suitable for building from thermoplastic polymers or metals with low temperature of melting. It is widely available because of low price of the device and cost per print. The thermoplastic material is shaped like a filament and it becomes pliable in a heated nozzle. After softening, the material is injected on the surface of a table where it is cooled under thermoplastic temperature. In fused deposition modeling, the nozzle or table are moved and table are moved using servo motors. Fused deposition modeling 3D printing is probably the most widespread because of the low price of thermoplastic materials available as filaments and low price of printer devices. Many thermoplastic materials are available and suitable for fused deposition printing. The table shows the materials which may be introduced as the polymers. We can see there, for instance, styrene, polylactic acid, and nylon. Stereolithography is technique also known as photosolidification modeling. The technique combines a photopolymer as material and a laser as a source of light. A photopolymer table is placed in a vat with a submerged working table. The surface of the table is illuminated by a light beam and shape of the fabricated object is ensured by the two-dimensional movement of the table. An ultraviolet laser is the most common source for induction of the photopolymerization during printing. Ultraviolet light, polymerizable methacrylate, uranet acrylate, epoxide resin, epoxy acrylate, and the others in the table are applicable in stereolithography. Stereolithography typically provides better accuracy of print than fused deposition modeling. On the other hand, commercially available devices are typically more expensive. In short, I'd like to highlight that the advent of microplasma in analytical chemistry represents a significant advancement in the field, offering miniature sources for elemental analysis. These miniature plasmas provide several advantages, including portability, low power consumption, and the ability to analyze samples in situ or in remote locations. They have shown promising capabilities in detecting and quantifying trace elements with high sensitivity and selectivity. Furthermore, their compatibility with various spectroscopic techniques enhance their versatility and applicability across different analytical scenarios. There is also possibility to print miniaturized systems using 3D printer. As research in this area continues to evolve, it is evident that microplasma technology holds immense potential for revolutionizing elemental analysis, paving the way to more efficient and accessible analytical methodologies in the future. Here are references. And thank you for your attention, and feel free to ask questions on my presentation.
Okay, thank you, Angelika. We are hurry in a short of time, so maybe one question from the audience, please. So maybe I will ask one. To create plasma, a stable and continuous supply of gases maintaining the supercritic state is needed. How this method was solved in a portable setting, if Angelika is with us?
Yes, I'm present.
Okay.
Can you repeat?
To create plasma, a stable and continuous supply of gases maintaining the supercritic state is needed, right? So how this method was solved in the portable setting?
Because my research is also connected with miniaturization of plasma systems, and now I'm on very preliminary research. So in my case, I can answer on this question because I didn't start generate plasma in my systems, but in this case of this article, they obtain this very stable plasma, so therefore they can determine these elements.
Okay, fine, thank you. So now the third presentation will be provided by the Jonathan Cardoso from University of Coimbra, titled Deep Euclitic Solvents as Alternative Additives to Increase Carbon Dioxide per Nitrogen Selectivity in PES per SAPO34 Mixed Matrix Membranes. So please start the recording.
Hi, my name is Jonathan Cardoso, and I'm here to present a part of my PhD work about the influence of dipeutetic solvents in PES SAPO34 mixed matrix membranes for CO2 and N2 gas separation, oriented by Professor Zilling from Aveiro University, Paulo Brito from Polytechnic Institute of Bragança, and Licínio Ferreira from University of Coimbra. This presentation is divided into introduction, experimental methodology, results and discussion, conclusion, and funding and acknowledgements. Although PSA, TSA, and cryogenic distillation are mostly used industrially in CO2 separation, the use of membranes in gas separation continues to be an attractive process since it provides lower energy consumption, is modelable, requires smaller processing area, does not require regeneration of solvents, and can be connected to traditional process. Mixed matrix membranes have a wide combination of polymers and zeolites have inorganic crystalline structures with uniform sized pores of molecular dimension. The silico-aluminum phosphates appears as one of the main materials in mixed matrix membranes for gas separation because of its pore size is near the connect diameter of gases like hydrogen, CO2, nitrogen, carbon monoxide, and methane. Those materials have been used as dispersed phase in polymeric matrix, aiming to obtain membranes with higher thermal stability and higher selectivity than polymeric membranes, as presented in Robertson upper bound, combining characteristics of organic membranes for higher permeability and inorganic fillers for higher stability and selectivity. However, the use of fillers in mixed matrix membranes still presents problems due to the compatibility between the dispersed material and the dispersive media, which results in the presence of interfacial voids between the polymer and zeolite, creating non-selectivity defects that lowers the separation performance of the membrane. So choline chloride-based dipeutetic solvents are proposed to enhance the interaction between those components due its high CO2 absorption and dual nature. So SAPO34 zeolite was prepared following the molar composition as presented in this slide, using dry gel synthesis for 24-hour reaction time. This condition was established in a previous work, then characterized by PXRD to verify the obtained phase, LDS to assess the average crystal size, and N2 adsorption and desorption to evaluate the textural properties. The membranes were prepared following the mass composition as presented in the slide for neat PES, PES SAPO34 and PES SAPO34 dipeutetic solvent membranes. The amount of dipeutetic solvents were defined in 5%. The solutions were casted using a 0.4 millimeters knife and proceeded to a solvent removal process at 90 degrees for 8 hours and 200 degrees for 24 hours in vacuum. Moreover, the membranes were initially characterized by permeation tests using CO2 and nitrogen and SEM analysis. Gas permeation experiments were conducted using pure gases to determine their ideal permeability and selectivity. The permeability results were compared to each other in a unit called bearer, while the selectivity results represent the ratio between the permeability of two distinct pure gases. A schematic representation of the permeation system is presented. The membranes were cut into 18 millimeters disc and mounted on a steel permeation cell and tested at feed pressure of 10 bar at room temperature. The adsorption-desorption nitrogen isotherm shows a type 1 isotherm. The textural properties show that the samples prepared have a large amount of micropores with a high surface area, BAT area, and with the volume of microporous compared to others in literature. The PXRD presented high purity in 24 hours reaction time according to the standard used as comparison criteria. The average particle size obtained by LDS analysis was 150 nanometers. This size is suitable to be used as fillers for the mixed matrix membranes without causing a high agglomerate of crystals promoted by crystals lower than 100 nanometers. When comparing the neat PES and PES SAPO34 membranes, reduction in CO2 and N2 permeability is noted. This fact is due to the addition of the zeolites with molecular sieving properties that cause an increase in selectivity due to their intrinsic properties and by changing the membrane morphology. Moreover, the permeability of gases decreased mainly by the increase in chain rigidification due to addition of the zeolite, blockage of zeolite pores, and extended diffusion pathways of penetrants through the membranes. The incorporation of different dipeutetic solvents in the PES SAPO34 membranes resulted in different influences of the CO2 and N2 separation. Choline chloride urea improved the CO2 and N2 selectivity, causing a reduction in permeability, which mainly could be resulted by increased pore blockage. Choline chloride glycerol improved the separation and increased the permeability, mainly CO2, which can be explained by the increased interaction of polyether sulfone and SAPO34 promoted by this dipeutetic solvent. Choline chloride ethylenamine inclusion reduced the CO2 and N2 selectivity and gases permeability, leading to a less useful membrane. So the membrane using choline chloride glycerol presented the best results as presented in the Robinson graph. SEM analysis of neat PES membranes, cross-section, and surface presents a dense membrane with no imperfection and similar homogeneity through the membrane. PES SAPO34 membrane's cross-section shows agglomeration of crystals along the polymeric matrix that could allow gases to permeate through the interface of crystals. However, in the present image, it can be noticed that the polymer covers all particles, even in the interface between them. This is crucial to mitigate performance drops and can be associated with the drying temperature used close to the TG of the material. SEM analysis of the PES SAPO34 choline chloride glycerol with 5%. The cross-section shows more dispersed crystals in the polymeric matrix, while the surface shows a similar pattern as presented in the PES SAPO34 membranes. With that, the conclusions we have is that 5% of different choline chloride-based dipeptidic solvents were using during membrane preparation to compare the influence of choline chloride urea, choline chloride glycerol, and choline chloride ethylenamine in mixed matrix membranes. The results showed that PES SAPO34 membranes containing 5% of choline chloride glycerol presented the highest separation performance. The use of dipeptidic solvents does not affect the general structure of the polymer, however helps to diminish zeolite agglomeration during the cast and drying phases, as can be observed by SEM analysis. I would like to thank the financial support provided by FCT and by University of Coimbra, University of Aveiro, and Polytechnic Institute of Bragança. Thank you for your attention.
Okay, perfect. Thank you, Jonathan. So we have already a question in the chat. Kamil Klinkowski asks, can the presented membranes be used as alternatives to biological membranes? So, Jonathan?
Yes, I'm here.
Did you hear the question?
Sorry, could you repeat if
Of course. Can the presented membranes be used as alternatives to biological membranes?
No. At least this is not what we intend with this study. We are more focused on gas separation, especially pre-combustion, post-combustion gases. So this is our main focus now.
Okay. So there's any other questions from the audience? Okay, Dominika Kapuścińska, please.
I have one question. How can the membrane you presented be used additionally? Please give some practical examples.
For example, now we are testing those membranes to try to separate CO2 and methane. We could use, for example, to increase the methane content, removing the CO2 from some bioreactors that generate biogas. So we could have a higher content of methane in the final product, for example, than the initial biogas generated by those bioreactors. So this could be one practical use of those membranes.
Okay, thank you.
You're welcome.
Thank you very much. So another questions from the audience? Okay, I can't see anything, so I think we can go further. So next one presentation is from Justyna Nowicka from Wrocław University of Science and Technology, titled Selective Cobalt Recovery via Electrodialysis. So please start the recording.
Good morning. My name is Justyna Nowicka. I am a PhD student at Wrocław University of Science and Technology, at the Faculty of Chemistry. Today I am going to introduce the process I am currently researching and its specific application in recovering cobalt from waste lithium-ion batteries. Throughout this presentation, we will explore whether batteries can serve as a secondary source of metals and the importance of such a source. Then, I will delve into my approach for extracting cobalt from used batteries, followed by a brief overview of future research directions. Lithium-ion batteries are ubiquitous nowadays. Chances are each household has a dozen times more batteries than residents. With the rise of electric vehicles and alternative energy, their production is expected to skyrocket. However, as batteries wear out, we face the challenge of preventing them from ending up in landfills and harming the environment. Let's explore whether they can be transformed into a new source of raw materials. Ion batteries contain various metals: lithium, cobalt, nickel, and manganese in the cathode material, aluminium and copper in current collectors, and steel in the casing. Of course, they can become a new source of raw materials, implementing the idea of sustainable development. The process of retrieving raw materials from scattered secondary sources is referred to as urban mining. But hold on, do we even need to use such complicated and demanding methods? According to the European Union's study on critical raw materials, we assess a material's criticality based on its supply risk, which describes problems with extraction from natural sources, and economic importance. The metals in question all fall well outside the safe zone. Importantly, their primary sources are often in remote or politically unstable regions. To extract metals from batteries, we first need to obtain an intermediate material known as black mass. This involves fully discharging and disassembling the batteries, then separating and grinding their components. Thermal and chemical treatments break down the cathode material's crystalline structure, enabling metal separation. The resulting black mass serves as a feed for the separation processes. The process I have been studying for this purpose is called electrodialysis. Two factors have to be present in every separation process. One causes the particles to move, the other separates them. In electrodialysis, movement of ions is included by an external electric field that opposes the concentration gradient, whereas the partitioning factor is a membrane. In this case, a cation exchange membrane, which means that it only allows cations to pass through. My aim was to develop a membrane selective for cobalt ions, yielding a concentrated cobalt solution and a dilute mix of other metals for further processing. The advantages of using this process are its low energy consumption and mild conditions, Moreover, it can be carried continuously, and some of used energy can be recovered from concentration gradient. It might have great influence on materials supply chains by closing technological loops. Most of my research on electrodialysis was conducted during my master's studies, and it's now being continued by other researchers. During my research, I had attempts to synthesize chelating-cation exchange membranes selective for cobalt ions. I then carried out material tests of the membranes and examined their absorption capacity for metal ions. Finally, I conducted process tests using the obtained membranes in the electrodialysis process, the results of which I would like to present. Initial tests involved single-component cobalt and lithium solutions, revealing promisingly low transport of lithium ions. In mixed solutions, the membrane exhibited clear selectivity for cobalt, especially in the presence of manganese ions. I also tested various current conditions, membranes, and ion concentrations. Take care when reading these charts, as I had to use different axis units for readability. I have drawn three key conclusions from my research. Firstly, selectivity of the membrane towards cobalt ions has been achieved, which encourages further research. Secondly, the obtained recovery ratio of cobalt was low. However, the literature reports much higher recovery ratios for similar methods, suggesting room for improvement. And thirdly, manganese visibly affects the transport of ions in the system. I think it's worth exploring. This marks just the beginning of my work on electromembrane processes. Let me briefly explore what lies ahead. Further efforts will focus on obtaining selective membranes for other metal ions, exploring alternative membrane synthesis methods, and optimizing processes to enhance recovery ratios. I am also working on capacitive deionization and hope to test it on industrial black mass. Thank you all for your attention. I am open to any questions, comments, or ideas you may have. Please feel free to reach out to me.
Okay, Justyna, thank you very much. So, is there any questions from the audience to Justyna? Okay, I can see that someone writes something in the chat. So the question is, what are the advantages of the presented membranes compared to the classic ones used?
Well, the greatest advantage in comparison with common used membranes is that, for example, commercial membranes are not selective. Well, they are selective by the charge. It means that they pass through only cations, for example, Maybe the more selective they pass through only monovalent cations, but there are no commercial membranes selective for specific element.
Okay. Any other questions from the audience? Okay, so maybe I will ask the question, is this method has any limitations? What are they?
Well, two limitations I met in my studies. First is that it is hard to scale up the process because the method of the membrane modification is complicated. And second is more like technological problem because the water uptake of my membranes is slightly big. It contains a lot of water and has to be stored only in water. And due to this, the stability of dimensions is poor, what can affect the process, especially in greater scale.
Okay, thank you. And I see that Camille asked another question out in the chat. So what are the detection limits and are you aware of the measurement error?
Yes, I am. This is one of my problem during the process study because I had to use greater concentration of lithium to prove that it is not passing through. My electrode for lithium has a limit of detection about 3 Siemens, and I had to just switch my study to one step greater concentration using about 0.1 mole per liter.
Okay, thank you. So we are waiting for other questions to Justyna. Okay, I can't see any more. Thank you, Justyna, for your presentation and for asking for your answers. And we can go for another presentation, the last one in our second session. It's from Marta Siol from Warsaw University of Life Sciences. And Marta will show us the application of chromatographic and thermal methods to study fatty acids composition, positional distribution, and melting profile as important factors characterizing pomegranate seed oils. So, please start the recording.
Hello, my name is Marta Siol. I'm from Warsaw University of Life Sciences, Institute of Food Sciences, Department of Chemistry. And I have got the pleasure to talk to you about application of chromatographic and thermal methods to study fatty acid composition, positional distribution, and melting profile as important factors characterizing pomegranate seed oil. Fruit processing generates a significant amount of waste in the form of pomace, which accounts for 10 to 45% by weight of the raw material. They contain pulp residues, seeds, and peels. During processing, the seeds are removed, becoming a byproduct that usually remains unused, although research indicates that this waste can be a source of nutrients and bioactive compounds that when isolated can be used, which is in line with the European Union's closed-loop policy, sustainable development goals, and the idea of zero or less waste. Pomegranates are used in the food industry, example for the production of juices, jellies, jams, wine, as well as for flavoring and coloring drinks. This fruit is rich in seeds, which are removed during processing, becoming a byproduct that could be reused. Pomegranate seeds constitute up to 15% of the weight of the entire fruit, and the amount of the oil in the seeds range from 12 to 20% of the dry weight. Oil from pomegranate seeds has 65 to 80% conjugated fatty acids, tocopherols and phytosterols. This oil has also strong anti-inflammatory and anti-edema properties and high antioxidant activity. Its distinctive feature is the punicic acid, which show anti-diabetic, anti-proliferative, and anti-cancer effects. Pomegranate seed oil supplementation counteracts obesity and insulin resistance caused by a high-fat diet. Punicic acid is a compound that could be potentially be used to treat type 2 diabetes. The aim of the study was to assess the quality of commercial pomegranate seed oil. The material for the study was commercial pomegranate seed oil, and the oil was stored through the study period in the dark glass bottles. Using the titration method, the acid value, which determines the content of free fatty acids in 1 gram of fat, as well as the peroxide value, which indicates fat oxidation, were determined in the tested oil. The composition of fatty acids was determined using gas chromatography in accordance with ISO standards. The tested oil was subjected to enzymatic hydrolysis using pancreatic lipase specific for ester bonds, taking the external position. Oil melting curves were obtained using a differential scanning colorimeter. The results of the analysis was a DSC diagram for the tested oil, showing the dependence of heat flow on temperature. The degree of fat hydrolysis, so the content of free fatty acids, is determined by the acid value, which determines how many milligrams of potassium hydroxide are needed to neutralize free fatty acids present in 1 gram of the tested fat. According to the requirements contained in Codex Alimentarius, in cold-pressed oil, the acid value should not exceed 4 mg potassium hydroxide per 1 gram of fat. The average of the acid value of the tested oil was 2.72 mg of potassium hydroxide per 1 gram of fat. The peroxide value is a measure of the peroxide content and determines the degree of oxidation or randicity of the fat. The ISO standard specifies the peroxide value in cold-pressed oils, which should not exceed 10 milli-equivalent of oxygen per kilogram, and according to Codex Alimentarius, the peroxide value should not exceed 15 milli-equivalent of oxygen per kilogram. The peroxide value of the tested oil after opening was 6.40 milli-equivalent of oxygen per kilogram, so it was within the range of both standards. The figure shows the percentage composition of fatty acids in pomegranate seed oil divided into groups: polyunsaturated fatty acids, monounsaturated fatty acids, saturated fatty acids, punicic acid, and others. The largest share in pomegranate seed oil was punicic acid. The content of monounsaturated fatty acids was 8.45%. The monoenic acid in pomegranate seeds oil consisted mainly of oleic acid and a small amount of godinic acid. Linolenic acid, alpha-linolenic acid, and eicosapentaenoic acid were polyunsaturated fatty acids contained in pomegranate seed oil, with the largest share in this group is linolenic acid. Saturated fatty acids in pomegranate seeds oils accounted for 7.25%, including palmitic and stearic acid, as well as trace amounts of arachidinic acid. Not only the profile of fatty acids, but also the structure of triacylglycerols significantly affects lipid metabolism in the human body. If saturated long-chain fatty acids are esterified in the external position, they are less digestible than unsaturated fatty acids in this position. During digestion, pancreatic lipase releases saturated acids from external sources, which tend to form calcium salts and are less well absorbed by the body. The same acids esterified in the internal position do not form calcium salts, and their absorption by the body is much better. Analyzing the composition of selected fatty acids in pomegranate seed oil, it was observed that punicic acid was the most abundant in the internal position, the share of which is in position was only 24%. The smallest amount in the internal position in the tested oil was stearic acid. Oleic and linolenic acid prefer the internal position of triacylglycerols, as their share in the position was over 43%. The share of palmitic acid in the SN2 tag position was 42.81%, which proves the even distribution of this acid between the internal and external position of triacylglycerols. Melting and crystallization are two physical transformations used to characterize the thermal behavior of lipid samples. The different composition of triacylglycerols influences the melting and crystallization curves of the tested lipids. The measurement results are presented in form of DSC curves, showing the amount of heat exchanged by the sample per unit of time as a function of temperature. On melting curves for the oil, two endometric peaks appear, the first in the temperature range from -80 degrees Celsius to -55 degrees Celsius, and the second in the range -54 degrees Celsius to -13 degrees Celsius. The minimum temperatures for them are -73 degrees Celsius and -31 degrees Celsius. Analyzing the presented DSC diagrams, it can be assumed that the first peak corresponds to the melting of triacylglycerols, in which polyunsaturated fatty acids predominate, which are most abundant in this oil. And the second peak corresponds to the melting of triacylglycerols, in which monounsaturated acids predominate. Just before I finish, let me summarize the main points. So, the tested oil, despite the high content of polyunsaturated fatty acids, was characterized by good oxidative and hydrolytic stability, as the acid and peroxide value did not exceed the permitted standards. Punicic acid was the main fatty acid found in pomegranate seed pressed oil. Pomegranate seed oil is a source of monounsaturated and polyunsaturated fatty acids. In pomegranate seed oil, linolenic acid and oleic acid were mainly found in the internal position of triacylglycerols, while punicic acid was mainly found in the external position. The DSC melting curves of oil were characterized by two main endometric peaks. The first peak corresponds to the melting of triacylglycerols, in which polyunsaturated fatty acids predominate, and the second peak corresponds to the melting of triacylglycerols, in which monounsaturated acids predominate. References. And this is all. Thank you so much for your attention.
Thank you, Marta. Is there any questions from the audience? I see that someone raised. Okay, so maybe Adriana Kosciuruk, she raised a hand. So please, Adriana.
Okay, hello. I have one question. Does any manufacturer produce pomegranate seed oil, or it is your own idea?
In Poland, no, but in Croatia, it's a lot of manufacturers where produce pomegranate oils and pomegranate juice. In Poland, no.
Okay, thank you.
Thank you very much. So I will read the comment from the chat, the question from the chat. Is pomegranate seed oil used in the food industry?
Currently, pomegranate seed oil is usually used for cosmetic purpose, but looking at its fatty acid composition and fatty acid distribution, it's assumed that it could be used as a functional distinctive or addictive, and dietary supplement. But the other hand, its oxidative stability is low, what's suggesting that it would be good to mix pomegranate seed oil with other oils. Also, a good solution would be to use a microencapsulation process to encapsulate this oil.
Okay, thank you, Marta. So there are any other questions from the audience? No one's raising a hand, but I gotta write the question. Triacylglycerols are the main class of lipids in the total lipid pool in the analyzed extracts.
Yes, are the main class of lipids.
Simple questions to answer. So we are still waiting for other questions. Is anyone interested in asking? Okay, I think I don't see any more. So thank you, Marta, very much for your presentation and answers once again. And now I think it's to have that's the end of our second oral session. I would like to thank to our speakers for interesting presentations and to our audience for the questions. Now we have a little break and we meet again at 12 o'clock for third oral presentation. Thank you for today and see you soon.
Okay, before we started, I have a question. Do you hear me and do you see me?
Yes.
Okay, thank you. So, hello and welcome everyone to our third presentation session. My name is Krzysztof Polaczek and I will have the pleasure to conducting this and the next presentation and the next session for you. Like the previous section, this one will be based on the recorded video of our speakers. Now we will see the first presentation, which was made by Karina Lenard from Wrocław University of Science and Technology, and the title will be Innovative Method of Juices Sterilization with the use of cold atmospheric plasma.
Hello, it is a great pleasure for me to participate in this conference. My name is Karina Lenard and I am a first-year PhD student at Wrocław University of Science and Technology. Today I would like to share results of research connected with the topic of innovative method of juices sterilization with the use of cold atmospheric plasma. Currently, there is a problem with food spoiling too quickly. So this causes its excessive waste. And the aim of the research was to initially assess the possibility of using cold atmospheric plasma for sterilization of selected freshly squeezed juices. It was citrus juices such as orange, mandarin, and grapefruit. And the order was to sterilize them. To prepare the juices, the fruits were, of course, washed with deionized water. The juicer was disinfected and the plasma system was sterilized. After squeezing the juice from approximately 2 kilograms of fruits, it was sedimented and decanted and then treated with cold atmospheric plasma. To treat juices with cold atmospheric plasma, a PM-RF APGD, pulse modulated radiofrequency atmospheric pressure glow discharge system was used. On the slide, you can see the photography of this system, and it consists of a peristaltic pump, a radiofrequency power supply, and a discharge chamber. The pump transports the sample through a silicone hose to a quartz capillary surrounded by a graphite tube. This tube is connected to the power supply using a platinum wire. The flowing liquid constitutes the electrode. The second electrode is made of tungsten, and plasma is generated between these electrodes. The sample after treatment is collected in a separate vessel, and appropriate operating parameters should be selected for the treatment of juice samples. So these parameters such as flow rate, frequency, and duty cycle conditions. And for this purpose, samples were treated with cold atmospheric pressure plasma under 15 different selected conditions. Okay, but now how it's possible that juices can be sterilized potentially as a result of the action of cold atmospheric plasma? So during no thermal plasma juices treatment, reactive oxygen and nitrogen species are produced, and this reactive oxygen and nitrogen species might putatively reduce the number of microorganisms naturally occurring in the analyzed juice. And to check the changes in the physicochemical properties of juices as a result of their treatment with cold atmospheric pressure plasma, of course, a number of analyses were performed. These were pH, conductivity, determination of saccharide content, so Brix measurements, estimation of the concentration of ascorbic acid, and determination of the number of colony forming units of the microorganisms. The results of the analysis were as follows. So no significant change in pH and determination of saccharide content was observed as a result of treatment, and an increase in the conductivity of juices after plasma treatment was observed, which is most likely caused by the formation of reactive oxygen and nitrogen species, such as, for example, nitrates and nitrites. And a slight reduction in the concentration of ascorbic acid was observed, and a high percentage reduction of the number of colony forming units of the microorganisms in juices which were treated with cold atmospheric pressure plasma. And what was quite important to find optimal conditions for juice treatment, multivariative optimization of working parameters of the applied system was performed, including duty cycle, frequency, and sample flow. And for this purpose, the treatment parameters were selected. So it was pH, which was minimum, conductivity was maximum, determination of saccharide content bricks measurement was maximum, estimation of the concentration of ascorbic acid was also maximum, and reduction percentage of colony forming units of microorganisms was also maximum. And now to conclude, the PMRF APGT plasma system can be successfully used for sterilization of juices due to the high percentages achieved in reducing the number of colony-forming units of the microorganisms in juices treated with plasma, and at the same time, no significant decrease in the concentration of ascorbic acid in samples was observed. So it's quite beneficial for potential applications, for example, in the food industry. And this research was financed by National Science Center Poland, Opus 2023 project, which is granted to Professor Anna Dimitrowicz and titled Opracowanie nowych procesów wytwarzania oraz sterylizacji funkcjonalnych napojów roślinnych z zastosowaniem technologii zimnej plazmy atmosferycznej. Thank you very much for your attention.
Okay, so many thanks for your presentation. Karina, are you with us?
Yes, hello.
Okay, hello. So now is the time to ask some questions to our speaker. So, okay, I don't see any questions so far. So maybe I will ask some question. Is there any limitation of this method? Which I mean, there are some juices which cannot be sterilized by atmospheric plasma.
At this moment, we are trying to use this method for citrus juices and for apple juices, but I'm sure that we can also use this plasma method for different types of juices. You can find many articles about it. But maybe in the future we will also check some other beverages, not only juices.
Okay, thank you. I see there's some question on our chat, so maybe I read it. What is the financial cost of using cold plasma for sterilization?
The financial cost of using plasma for sterilization of juices hasn't been yet checked, but I believe that when this method makes that the juices can be, it can make the shelf life of the juices much longer. It is it can be used in the food industry. I believe that the estimation of the financial cost is also important. So maybe we will make some analysis of it in the future.
Okay, thank you. I think we still have time for some questions. So please, Professor Axman.
You said that this kind of sterilization do not influence ascorbic acid content in the juices. That's of course very good, but I'm wondering if it has some influence on parameters that are important for consumers, for example, taste of the juice or even color or something like this.
When we talk about the taste, we didn't try the taste of these juices yet. I think it can be do in the future, yes, after many other tests on these juices. When we talk about color, the color of juices can a little bit change, but it's just a little change in color.
Thank you.
Thank you.
Okay, is there any other question for our speaker? So, if it's not, thank you for very interest. Oh, okay, sorry, it's the one question. We still have time. Have you tried to respond surface methodology to most relevant set of conditions without doing thousands of experiments to check? It's a question for Wojciech Snoch.
No, we didn't. Please give me some information, more information about it. Maybe after session, I would like to hear more about this surface methodology.
Okay, Wojciech, I encourage you to contact with Karina after the conference. So now, is there no question? Thank you again for
Thank you very much.
And now we will hear the presentation of Klara Żbik from Warsaw University of Life Science. And the title of this presentation will be Method for the Rapid Identification of the Volatile Organic Compounds Profile of, Zadurella tomatoes stored under different conditions.
I'm so glad I could join today's conference and show you the results of our work. I'm Klara Żbik from the Warsaw University of Life Sciences, and with Elżbieta Górska-Horprzyczak and Weronika Siedlecka, we carried out research and want to present you the exciting and rapid method for identifying the volatile organic compounds in tomato samples. So let's begin with some introductory pieces of information. Okay, so first one. You can smell the aroma of different food products thanks to small molecules that easily evaporate from the matrix and react with olfactory receptors that are mostly placed in the nose and throat. All types of food, animal origin and plant-based, emit these molecules that called volatile organic compounds, VOC in shortcut. Today I'm talking about our experiment on tomatoes, so let's focus more on plant origin VOCs. They are an essential part of plant metabolism, like language for them. Thanks to VOC, plants can signal to each other, repel pests, and attract bees and other pollinators. So this is a massive group of molecules that differ in weight, structures, and of course ways that our olfactory receptors receive them. In the electronic nose method that we used, very important feature is that you can see in the first cycle a low boiling point. What's an electronic nose in general? It's a set of equipment you can see in this picture. Here we have an autosampler that takes a vial that is waiting in this trace, and autosampler slides on this rail. Next, put the vial in the incubation unit for a set time. We can manipulate all parameters like time, temperature, and vial rotation in the incubator. After that, the Amsel sampler takes a headspace sample from the vial and injects it into chromatography columns with a 10x strap. An essential part of this set that you can't see here is that the computer with software helps with VOC identification and chromatogram interpretation. At the end of the process, we receive the profile, which is a fingerprint of the sample, something like that, and analysis of one sample can last, depending of its nature and nets we have, for several dozen minutes. Fingerprints can help determine food products features like changes during shelf life and processing, and can be beneficial in finding contamination and frauds. Knowing our product's proper VOC profile, its fingerprint, we can quickly notice deviation from the norm and say that something is wrong with our sample, or maybe it's all right. We choose tomatoes for our research because they are not the first product we can link with a strong aroma, but I think we always wait for that because it's one of the most essential parts of this vegetable. So tomatoes are very delicate due to their high water content and are sensitive to storage conditions. Also, they are rich in valuable bioactive compounds, so preserving them in the best and most attractive condition is essential for industry and of course for consumers. After this short introduction, I want to say that the research aimed to determine change in the VOC profile of Zadurella tomatoes during storage in different conditions using the ENOS method. We used less popular tomatoes grown near Warsaw using a conventional method. Zadurella is a red medium fruit tomato, and the fruits used in this experiment at the beginning were in the 5th maturity stage. That means it was light red. You can see exactly tomatoes in these pictures. Next, we divided tomatoes into 3 groups, stored at 4 degrees Celsius, so-called cold storage. The second group was in 10 degrees, and this is recommended temperature for tomatoes to prevent chilling injury. And the last one was stored at room temperature. The experiment lasted 11 days, and measurements were carried out on the first day, second, third, fourth, seventh, and 11th day. And this is our procedure for every measurement day. So hand-shredded samples were weighted in vials in 3 replications. Next, we used electronic nose equipment, Heracles, that you sent before, with 2 column, polar and slightly polar, with 2 flame ionized detectors. I will show you more details in a minute about exactly ENOS methodology. The last step was to identify the VOC profile using software and prepare PCA analysis for better results visualization. And here I want to show you more details about these parameters for our INOs. To achieve the best results, you have to carefully choose at what temperature the sample will give off the most of VOC without negatively affecting them. So you can choose too high temperature or too low for the best results. How long the procedure should take, it's also your choice and very important. And the parameters of chromatography analysis to separate all VOC as best is possible. And to achieve this, you must rely on, of course, literature and carry out many pilot measurements until the results that you will getting will be satisfactory for you and for your sample type. Okay, so here are our results. I want to show you. In the first column are every detected VOC in samples from day 0 to 11. The next column is this one from day 11 exactly for tomatoes cool stored. The next one is for this stored in recommended temperature, and the last one was for this one in room temperature. Now you can see that samples differ depending on the day of measurements and storage conditions. Methanol and acetone that you have in red are compounds that negatively affect the aroma of tomatoes because they are sharp and unpleasant for us. Tomato aroma quality depends on the proportion of VOC with favorable and unfavorable aromas. On the last day of storage, the content of compounds with positive effect on the aroma, like nerol, 2-phenolanthanol and 2-methylbutane, this one in bold, were highest at samples from room temperature and lowest for this from cool storage. In contrast, the level of compounds considered unfavorable to tomato aroma, red ones, methanol and propan 2,1 acetone, were highest when stored at 4 degrees and lowest at 20 degrees. So two fruits stored at room temperature, we can say, had a better aroma quality than this maintained at 4 degrees. And here you have PCA, Principal Component Analysis, one of the most popular methods used to model, compress, and visualize multidimensional data uses distances. This one is for tomato storage at 4 degrees. Here is day 0, and here is the last one. Every day, how you can see, differs significantly. During storage, the profile of tomato VOC changed very dynamically. Samples for day 7 were separated by the highest distance and in the opposite part of the graph compared to the samples from day 0, indicating significant difference in this composition. So here we have results for tomatoes stored at 10 degrees. The most significant changes in the profile of VOC were recorded compared to the last day as evidenced by the highest value of distance between day 0 and 11. You see. And the distribution of these samples in opposite parts of the graph. The last one is PCA result for samples stored at 20 degrees. Analyzing the chart, I can say that the most remarkable difference from the profile of volatile compounds from day 0 were characterized by the profile that we obtained on the 7th and 11th day of storage. 7 and 11. Previously, changes did not occur as dynamically as in the samples shown earlier, because you can see most of them is almost in one place. These are all the results I wanted to show you today. Now I want to briefly summarize what comes out of the research. This type of analysis can be a method to differentiate batches of tomatoes and of course different products. And firstly, the Zadurella aroma profile was characterized by 13 VOCs, and I can say that the best storage temperature for aroma preservation was 20 degrees, when the worst was 4 degrees. It worth noting that the profiles of tomatoes stored at 11 and 20 degrees were similar, but the second group, this one in room temperature, was more favorable. That's all for today. Thank you very much for your attention, and I'm glad that I could show you this presentation. I hope you enjoyed this exciting eNotes method, and if you have any questions, you can write to me. Thank you.
Okay, we are also thank you for your presentation. Now it's time for, I think, a quick question to our speaker. Yes, we have some question on the chat, so I will read it. What other applications the electronic nose may have? It's from Marta Siol.
Thank you for the question and your attention. I want to say that how I mentioned before, that you can use this method, for example, to detect food fraud. So you have to prepare a profile for your not fraud product, like for example, like olive oil. And next you can, thanks to this method, in few minutes, just check that your product that you bring to laboratory is exactly the same in his profile like this your standard. So just for example a clear olive oil, not this one fraud with, for example, different oil from just cheaper material. And you can do it exactly at the same method with meat, like mixed meats, beef with pork, for example. You can just check that in your part of meat grounded, it's more meat from beef or more from pork, so it's a type of food fraud too.
Thank you. Adrianna, you also have a question?
Hello, I have one question. What is the sensitivity of this method?
This method is very sensitive, but I don't want to talk exactly about limits, etc., because it's more like idea of qualitative analysis, not about exactly amounts of exactly, about this VOC. So you have to think about this method more about like fingerprint. Not everything is you can detect, but at the end you have something like picture that you can see, that you can say that I see this one. I see this one. It's like fingerprint. I don't want to talk about exactly limits and detection limits in this case.
Okay, thank you very much.
Thank you also. I see that we have another question from Katarzyna Wiktorska. Time is ready, so please, the quick question.
Okay, so maybe the simple answer yes or no will be enough. Maybe I didn't catch it, but is this very smart method somehow comparable with our scent? So how we smell the products?
I can say yes.
Okay, so I mean we can, because you said that there's a difference. For example, this after 2 or 4 days, or the 7 days was the most significant difference. But are we able to also detect this difference by our sensor, our nose?
Probably yes, because in the case of tomatoes, they are repairing very fast, so probably yes. But this is just an example in this method. So the best of all of that uses is for me in these cases when we can't say that with our noses, like when we just checking food frauds. So in this case, tomatoes, I can say yes, because distances on these graphs was
huge, distinct, yes.
But the best is, I think, when we are using it with these food frauds cases. Yeah.
Thank you.
So,
thank you.
Thank you.
Okay, once again, thank you very much for your presentation, and many thanks for the question from the audience. And now we will see the presentation made by Zuzanna Hirsz from University of Gdańsk, and the title was Polyphosphate granules serve as an alternative scaffold for lawn proteas during stress conditions.
Hello, my name is Zuzanna Hirsz and I am a PhD student working under the supervision of Professor Igor Konieczny in the Laboratory of Molecular Biology at the Intercollegiate Faculty of Biotechnology. The title of my today's presentation is Polyphosphate granules serve as an alternative scaffold for lawn protease during stress conditions. In the reality of the smallest organisms, such as bacteria, the balance among various molecules, it's not just a biological phenomenon, it's their survival strategy. In the face of the constantly changing environment, these microscopic organisms have mastered the art of maintaining stability to ensure their continued existence. One aspect of this stability is called proteostasis, which is the equilibrium between synthesized, degraded, and folded proteins in the cell. The main players, degraded proteins, are called proteases, and in the model organisms, the Escherichia coli, there are 4 main cytosolic proteases: ClipXP, ClipAP, ClipYQ, and LON. The activity of these proteases can be modulated, for example, when they interact with different molecules, such as DNA. Here, you can see the results of the in vitro proteolysis experiments, where the addition of DNA for these 2 proteases has the inhibitory effect. You can see that the band here representing the substrate is fainter in the sample without DNA, which means that there is less of it in the sample. Actually, but for these 2, the situation is different. The addition of DNA stimulates the activity of proteases, such as LON. LON is a well-studied, highly conserved protease that can be found in the archaea, bacteria, and in eukaryotes, and this is the protease that I decided to work with and investigate. Our laboratory also showed that the stimulation of LON activity by adding DNA is not exclusive for the TRFA protein, but also for different replication initiation proteins, such as bacteriophage lambda O protein or REP-E. LON protease is composed of 3 main domains: the N-terminal domain, the AAA+ domain, and the protease domain. 6 monomers of lon can form a hexamer, and 2 hexamers can form a dodecamer. Our group showed that besides the main assumption that lon is a cytosolic protease, almost all of the cellular lon in vivo is localized within the nucleoid, in the DNA-containing fraction. Our group also selected the amino acids that are responsible for the DNA binding of lon protease, and we used the SPR to confirm that. You can see here that the lon wild type can interact with the DNA, and here that the mutant that has the mutation in the selected amino acids lost the ability to bind the DNA. In vivo experiments showed that during amino acid starvation, the intracellular level of lon protease rises. So the increasing level of lon is one of the many responses to stress. To the stress, survival also has been linked in an enigmatic polymer called polyphosphate. Polyphosphate, which in short is called poly-P, is an inorganic phosphate polymer of hundreds of phosphate that can be synthesized in bacteria by the action of the enzyme poly-P kinase and can be later degraded by the enzyme poly-P exon phosphatase. A variety of different stress conditions triggered poly-P synthesis, such as starvation or oxidative stress. And despite the fact that poly-P is a chain polymer, in cells it can form granules, which in Pseudomonas aeruginosa can be seen using electron microscopy. Our group also showed that lon not only can interact with DNA, but also with polyphosphate. What is interesting is that the substrate for lon, the DNA A replication initiation protein, also can bind to the polyphosphate. What is even more interesting is that in the mutants which cannot synthesize poly-P, the DNA A protein is not degraded by lon. When you look at the higher panel here, you can see that in the wild type, the level of DNA A drops during stress, where in the mutant lacking polyphosphate, this level is stable. Our hypothesis is that polyphosphate can serve as an alternative scaffold to DNA for proteins. During stress, when polyphosphate is synthesized, the DNA binding proteins change the scaffold from DNA to the poly-P. Poly-P then creates a crowd of lon and substrates for efficient degradation. So the aim of my project is to investigate how stress affects the dynamics of lon protease in the context of its intracellular localization, concentration, and its interaction with poly-P. So for the determination of intracellular localization, I had to use fluorescently labeled lon protease. The label of choice is M-scarlet, red fluorescent protein, and I used 4 different variants of that recombinant protein. Those variants differ in linker between lon and M-scarlet, and I used variant without the linker, 2 variants with the flexible linkers, and 1 variant with the rigid linker. All contained fluorescently labeled proteins exhibited fluorescence, and I purified them to analyze their activity because I wanted for the recombinant protein to be the most similar to that of lon wild type in case of its proteolytic activity. I determined proteolytic activity in vitro of purified proteins and compared them to that of lon wild type. I used 2 substrates, the casein and TRFA initiation replication protein, which I showed at the beginning that the addition of DNA stimulates its degradation. Based on that results, I chose the variant with the flexible linker as the most similar to that of lon wild type. As for the intracellular localization of lon, I wanted to obtain the E. coli strain, which has the fusion of the lon gene with the M-scarlet fluorescent protein gene under the control of its native promoter, which is located on the chromosome. To obtain that, I used CRISPR-Cas9 assisted recombinering, which is based on the homologous recombination mediated by the bacteriophage lambda proteins, and it's coupled with the CRISPR-Cas9 system, where the Cas9 causes cell death through double-strand breaks. Therefore, it serves as a negative selection for the cells where the recombination did not occur. To use this system, I had to design and construct a vector that contains the donor DNA for recombination and a vector containing gRNA sequence that will guide the Cas9 to the sequence to create double-strand breaks. Then I had to optimize the whole process, which took me some time. And I obtained 3 E. coli strains with the lon M-scarlet fusion gene on the chromosome. 1 from the wild type, 1 lacking polyphosphate, and 1 that accumulates polyphosphate, where the last 2 can serve as the controls for the experiment. Used to obtain strains to analyze the localization of LON in the real-time manner using Onyx microfluidic system coupled with the fluorescence microscopy. This experiment starts with 25 minutes of growth in the normal conditions in rich medium, and after that I introduce stress by changing the medium to the minimal medium. In the second you will see that in normal conditions, LON is localized throughout the whole cell. It almost fills the cell. However, when the stress starts, the localization pattern of LON changes. It started to form a foci inside of the cell, as you can see here as the spots. So here are the cells in the normal conditions. They are normally dividing. And here is the point when the stress starts. The replication cycle is finished, cells stop dividing, and the foci are being formed inside of the cells. You can see them as the spots here and here. The formation of spots and the expansion of them is continuous throughout the whole stress to the point of the 3 hours, where the cells start to recover from the stress and they begin to divide again. Here you can see the DAPI staining of the chromosome and polyphosphate that was made in our laboratory. The DAPI staining of both molecules is possible because of the shift in emission wavelength of the dye when it interacts with poly-P. You can see that DNA fills out the whole cell, just like LON at the beginning of the movie on the previous slide, just before stress. Poly-P forms granules in the cell, which can be seen as spots along the cell. The next picture are different experiments where the chromosome is stained with the cyto dye, and for the visualization of poly-P, I used fluorescently labeled poly-P binding domain, which is the domain from the enzyme exopolyphosphatase. Here is the overlay of these two, and here is the picture from the stress with LON for comparison. I certainly can see the similarity in the localization pattern, which can suggest that during stress, LON localized with the polyphosphate granules. In the Escherichia coli mutant that accumulates polyphosphate, the poly-P granules can be visible, as you can see here. Here is the fluorescence of the LON abscariote protein and the overlay of both pictures. This suggests the confirmation of the hypothesis that when poly-P granules are formed, LON binds poly-P and changes the scaffold from the DNA. Using the Western blotting and the stoichiometric analysis, I investigated the intracellular level of LON protease during stress in minimal medium. I also determined the level of its substrate, the DNA replication initiation protein. You can see that as the stress progresses, the DNA level drops and the LON level rises and then also drops. This shows that the stress causes fluctuations in both protein and intracellular levels. So to sum up the results that I showed you today, when cells encounter stress, it impacts LON proteases intracellular localizations. During stress, polyphosphate granules are formed and LON localizes with them inside of the cell. And finally, stress causes fluctuations in intracellular level of LON protease. Thank you for your attention, and thanks for all the people that contributed to my project that I showed you today. Thank you.
Many thanks for the presentation. I think we have some time for one question, so does anyone want to ask something? Yes, I see the question on our chat from Dominika Kupuścińska. Do you have plan to investigate LON protease localization during other stress conditions like oxidative stress or presence of antibiotics?
Thank you for these questions. Can you hear me? Is everything okay?
Yes, we hear you.
Yes. I didn't plan to use different stresses because in the literature it was revealed that oxidative stress also
also makes the formation of the polyphosphate. So our hypothesis is that the formation of the polyphosphate is like this universal response to the stress conditions. However, it was showed that also in the, for example, in the oxidative stress, the polyphosphate is formed inside of the cells. So our hypothesis is that the localization of the lone protease will be very similar to that when we make the stress when we take the amino acids from the medium, so in the amino acid starvation.
Okay, thank you for the response. Once again, thank you for your presentation. And now we will see the presentation made by Izabela Wysocka from University of Białystok, and it will be about antioxidant compounds in human breast milk.
Welcome to my presentation. My name is Izabela Wysocka and I'm a first-year PhD student at the Doctoral School of Exact and Natural Sciences of the University of Białystok. My today's topic is antioxidant compounds in human breast milk. This presentation was prepared with help of my supervisor, Professor Edyta Nalewajko-Seliwoniuk. In this presentation, I would like to talk about maternal breast milk, free radicals, oxidative stress, and antioxidants. Then I'll give you information about antioxidant compounds in human milk. After that, I'll proceed to the preparation of the sample, extraction, and determination methods. In the end, I will sum up my presentation. Human breast milk is natural source of numerous nutrients. Its composition and bioavailability make maternal milk ideal food for infants. Breastfeeding newborns from the first days of life provides them with necessary nutrients and support healthy development. Maternal milk consists in about 87 to 88% from water, 7% of carbohydrates, 1% of protein, 3.8% of fat, and 0.2% of other important species like vitamins, minerals, antibodies, immune cells, hormones, and strains of bacteria. Mature milk contain up to 65 to 70 kilocalories per 100 milliliters of energy. Composition of human milk depends on many factors like mother's health and diet, mammary gland physiology, stage of lactation, and other environmental factors. Sunlight, pollution, long-term stress, and poor diet may be a reason of creation free radicals, molecular species with unpaired electron in atomic orbital. Many radicals are unstable and highly reactive. That's why they are capable of damaging cells and molecules, which cause oxidative stress. It's a condition of oxidative damage result when balance between free radicals and antioxidants content is staggered. In consequences, oxidative stress contributes to the occurrence of diseases such as Alzheimer's disease, Parkinson's disease, arteriosclerosis, and diabetes. Free radicals may also cause children illnesses like retinopathy of prematurity, intraventricular bleeding, or necrotizing enterocolitis. Antioxidants are substances that can donate electron to free radical and neutralize its capacity to damage. This creates an opportunity to stop destructive chain reaction and protects organism from negative influence of environment. Many of antioxidants can be found in food. That's why our diet should consist of vegetables, fruits, and other products rich in these compounds. First group of antioxidant compounds in human milk that I would like to present are polyphenols. They are metabolites of plants naturally occurring in fruits and vegetables. Their content in apple, pear, cherries, and berries may reach up to 200 to 300 milligrams per 100 grams of fresh weight. These substances are able to pass into breast milk and be assimilated by infants. Polyphenols are mostly known from having number of phenyl rings and structural elements that bind these rings together. Nowadays, more than 8,000 polyphenolic compounds have been identified in various plant species. The main groups of those compounds are polyphenolic acid, where representatives are caffeic acid, gallic acid, and ferulic acid. Flavonoids were represented by quercetin and catechin, and also stilbenes and lignans. Carotenoids are another important group of antioxidants that can be found in breast milk. They are red, orange, and yellow pigments synthesized by plants. Carotenoids regulate oxidative stress and inflammatory mediators, thus protecting against metabolic syndromes, cancer, neurodegenerative diseases, and photooxidative damage to the skin and eye. Their presence in human milk has significant value for visual and cognitive development of an infant. The major carotenoids contained in food are alpha-carotene, beta-carotene, beta-cryptoxanthin, lutein, zeaxanthin, and lycopene. The first three of them are called provitamin A carotenoids because they can be converted in vitamin A in the body. Lycopene is found abundantly in tomatoes and their related products, but can also be present in fruits such as watermelon and pink grapefruit. Papaya, peaches, and citrus fruits have significant levels of beta-cryptoxanthin. Lutein and zeaxanthin can be found in leafy green vegetables, such as broccoli or spinach. Tocopherols and tocotrienols are natural lipophilic antioxidant compounds. They are supplied in various amounts in a diet, mainly from vegetable oils, some oilseeds, and nuts. The main forms in the diet are alpha and gamma tocopherol due to the highest content in food products. Alpha tocopherol is the main form of vitamin E with the highest tissue concentration. The alpha forms of both tocopherols and tocotrienols are considered as the most metabolically active. Vitamin E is particularly essential for preterm neonates who have very low plasma levels of these compounds due to immature lipoprotein metabolism and low transport capacity. Vitamin E is involved in antioxidant activity and immunomodulation. Additionally, vitamin E is the most potent lipid peroxide radical scavenger, which protects tissues against chromosome damage and DNA oxidation. Here I present chemical structures of some of the tocohormones. Vitamin C is one of the best known antioxidants. It performs various functions and is involved in more than 300 processes in the human body. The biological roles of vitamin C are based on its reducting properties. Vitamin C is specifically required by human enzymes that synthesize and metabolize collagen, hormones such as adrenaline or serotonin, and carnitine, including enzymes that synthesize and modulate selected components of the nervous system, the conversion of cholesterol to bile acids, and the biosynthesis of connective tissue components, including elastin and the bone matrix. It also neutralizes free radicals and protects tissue from damage those species may cause. Currently, most of the daily intake of vitamin C comes from fruits and vegetables, which in many countries, unlike in the past, are available throughout the year. The richest source of vitamin C are rose hips, sea buckthorn, cacao plum, camu camu, and acerola. The reference intake for vitamin C is 75 milligrams per day for adult females, 85 milligrams per day for pregnant women, 120 milligrams per day for breastfeeding women, and around 40 to 50 milligrams per day for infants up to 12 months of age. Before we can even begin to determine antioxidant content in the human milk, we need to carry out a number of processes. The first of these is skimming to get rid of the fats contained in the sample. This is usually done by an hexane extraction. The next step is to separate the proteins from the matrix using, for example, ammonium sulfate. Then ascorbic acid is added to protect the antioxidant compounds from oxidation. The next step is the deconjugation of potential metabolites to aglycons using beta-glucocuronidase. After all these procedures, we can finally start to determine antioxidant compounds in breast milk. In the literature, various methods were used to extract and separate antioxidants from breast milk samples. Here I present some examples of them. For isolation of polyphenols, mainly liquid-liquid or solid-phase extractions were used. For the determination of this group of compounds, liquid chromatography with tandem mass spectrometry was the most frequently used method. Carotenoids were mostly extracted from human milk by liquid-liquid extraction or saponification. The most used methods of determination of this group of compounds was also liquid chromatography with various detectors, such as ultraviolet detector or diodate array detector. Extraction of tocochromanos was also mostly done by saponification or liquid-liquid extraction. To determine those compounds, also liquid chromatography was used with detectors like fluorescence detector or photodiodate array detector. Before the determination of vitamin C in the human milk, in majority of papers, researchers did not use any extraction technique. While for the determination, usually liquid chromatography with ultraviolet detection was used, or the titration with 2.6 dichlorophényl indophenyl were applied. On this slide, there are all abbreviations that was used in the previous tables. So, to sum up my presentation, human breast milk is the best natural source of various nutrients essential for proper growth and development of newborns. Antioxidants in maternal milk have significant impact on neutralizing free radicals, which prevents many diseases. Good source of antioxidant compounds are fresh vegetables, fruits, nuts, and edible oils. There are many methods to extract antioxidants from maternal milk, but unfortunately most of them are expensive and non-environmentally friendly. These facts provide an opportunity to develop new, greener extraction methods. Here are my references. And thank you for your attention. If there are any questions, I'll be happy to answer them.
Okay, thank you for your presentation. Yes, I see the first question. I think we still have time for one quick question. So Dominika Kapuścińska, please, can ask your question.
Okay, I have one question. Do you know how big the difference can be in the concentration of these compounds you mentioned in milk from different mother?
Hello, thank you for your question. Can you hear me?
Yes.
We hear you.
So the concentration of those compounds may vary because maternal milk is a very hard matrix to work on it, and the concentration of compounds depend on maternal health, maternal diseases. It can also depend on of the lactation step of the because in the first few days the compounds are more concentrated. But after few weeks of breastfeeding, those compounds are in smaller concentration. So it's really hard to tell how much of those compounds are in some mother's milk.
Okay, thank you very much.
Thank you for your question.
Yes, once again, we thank you for your presentation and for answering the question. And now it's time for the last presentation of this session. And it was made by Jacek Karwaszewski from Nicolaus Copernicus University in Toruń. And the title of his presentation is Bacterial Derived Polymer Polygamma glutamic acid, the key to survival of flowers of yellow lupin under drought stress.
Hi, my name is Jacek Karwaszewski. I represent the chair of plant physiology and biotechnology at the Nicolaus Copernicus University in Toruń. Today, I would like to show you the great potential of bacteria-derived polymer polygammic acid to counteract drought effect in yellow lupin. Poland and many other European countries are forced to import significant amounts of protein fillers despite having adequate area for their own producted. As you can see, soybean is most commonly cultivated for export, and the main countries responsible for its production are China, United States, as Latin America region. A solution to this problem could be cultivating of domestic protein crops to obtain substrate for the production of animal feed. In this context, the best choice is the legume family, such as broad beans, beans, or lupinus. The last one is among my research. There are many advantages of lupins as a crop. They produce high protein material for food and feed, and have low soil requirements given their ability to symbiosis with rhizobium bacteria with fixed atmospheric nitrogen. These legumes are extremely ecological species given since they improve soil and nitrogen. On the other hand, lupinus are strongly sensitive to drought. According to a report published by the European Drought Observatory and the Intergovernmental Panel on Climate Change, the increasing annual temperature amplitude in many regions of the world caused by anthropogenic climate change will result in the occurrence of drought. It has been recognized as one of the most devastating abiotic stress for plant productivity in countries where agriculture already fails to provide sufficiently fields to feed the local population. Even in medium-high risk regions such as Poland, drought is a serious problem. In yellow lupin, drought leads to premature and excessive abscission of flowers, and as a consequence, reduced number of formed seeds, which are source of protein. The decrease in legume yielding generates serious economic consequences. In general, organ separation is a controlled process of removing organ from plants. It takes place in a specific location named abscission zone, AZ. In the flower of lupin, AZ is located at the base of the pedicel and is composed of small, easy to distinguish cells, which are different than those presented in the distal and proximal parts. Once AZ is activated, AG by abiotic factors such as drought, the cell structure is modified significantly. Cells start to proliferate strongly and change in size and shape. The organ detachment process involved to the degradation of the middle lamella of cell walls between adjacent cells in the AZ that occurs through the action of many hydrolytic enzymes with changed cell wall composition. It has been proved that when plant is subjected to drought, the rhizosphere microorganism composition is strongly affected. These microorganisms can produce many plant protective substances that improve plant growth, development, and help to survive unfavorable conditions. One of these beneficial compounds is polygammic acid, which is naturally synthesized by soy bacteria of Bacillus family. However, despite the knowledge about the way of action of this component of plants, its impact on AZ functioning is still uncovered. That is why the aim of the presented research was to determine the influence of synthetic gamma PJ on the cell or related change take place in the flower AZ of yellow lupin cultivated under soil drought condition. Then it was possible to assess the potential of gamma PJ as an inhibitor of flower abscission and a compound that can increase lupin agronomic potential. These objectives were achieved through analysis related to examination of the flower AZ functioning and its activity level in the different experimental variants by light microscopy. investigation of the composition and distribution of pectin polysaccharides in the cell of the flower AZ using specific monoclonal antibodies. The research material used in the study was yellow lupin, Lupin sultaeus taper cultivar. The plants were grown under controlled condition, temperature of 22 degrees Celsius and photoperiodic on 60 hours of light and 8 hours of darkness in phytoton chambers. From germination until the end of the first month of cultivation, they were watered 3 times a week with the same amount of water determined by soil moisture content. To determine the impact of PJ, the following experimental variants were they distinguished. The first, positive control group, plants cultivated under optimal soil moisture content. Second, negative control group, plant with limited access to water, 25% soil water holding capacity for 2 weeks. PGA 100, plants subjected to drought condition, 25% soil water holding capacity for 2 weeks, and then treated with gamma PGA solution, 100 nanogram of liter. Histological material were acid fragments from flower pedicel with collected after 7 weeks of cultivation. Material was fixed for 12 hours, then it was rinsed dehydrogenated using a series of ethylene with DTT and placed it in solution with increased concentration of BMM, resin. The resin saturated material was placed in BEM capsules for polymerization conducted at minus 20 degrees Celsius from 96 hours under UV light. After that, the resin with the researcher material was removed from the capsules. Subsequently, using a microton, material was sliced into semi-thin sections, with then placed on microscope slides coated with an adhesive solution. After that, microscope slides containing semi-thin sections were rinsed with distilled water and then stained with solution of toluene and blue. Slides were left to dry, then observation and documentation of the results were performed using Zeiss Exciplan light microscope. For the immunodetection, microton slides containing semi-thin sections underwent the process of beam resin removal with acetone, water, and PBS. The resined tissue section were incubated with primary antibody solution against various types of the pectin, galactans, arabinians, or high-methylate aceticated monglactoronanas. Next, secondary antibodies conjuncted with fluorochrome were served. DAPI was applied to stain nuclei. The results of immunochemistry were observed using a fluorescent microscope. The results of histology analysis revealed that under optimal growth condition, flower AZ cells exhibited regular shapes and size, no visible cell divisions were observed, and no signal of AZ activation were noted in this experimental variant. When the plants were subjected to soil drought stress, cells of AZ exhibited irregular shapes and size. We observed visible cell divisions in the area pedicel base, especially near to the vascular bundles. The constituent cell of the AZ displayed a clear division line. Plasmolysis was evident. In 2 rounds, lupinus were subjected to soil drought stress, then treated with PGA 100 nanogram on liters. Cells of AZ exhibited regular shapes and size, no cell divisions were visible, and we haven't found signal of plasmolysis. AZ was clearly distinguishable against the background of surrounding cells and looked similar to inactivated one. In the case of this section, a higher number of cell divisions were visible compared to the control sample, but they are not organized as in plants not treated with PGA, gamma PGA. In the next step, we aimed to deeply analyze to pectin composition of AZ cells. Microscopic analysis of section stained with gene-7 antibodies against high-methylated homolacronans revealed that these components are highly abundant in AZ of flowers from drought-treated plants. As you can see on the field pictures of high magnification, especially cell walls were strongly labeled sporing, that this compartment in methylated pectins. On the other hand, when we analyze the section of AZ from flowers cultivated under drought condition and sickness and treatment with PGA, a lower intensity of signal was visible compared to stressed plants, indicating a decreased content of highly methylated pectins. The green signal was particularly emitted by cell walls. Microscopic analysis of AZ section stained with LM5 antibodies with detected beta-1.4-galactan. In this case, we did not observe a clear signal in the case of plant growing in the coordinates of pure drought. There was minimal emission observed, attributed to autofluorescence of the cell wall. In the situation change for plants treated with PGA, emission was presented with a significant increase observed compared to the variants not treated with PGA. Microscopic analysis of AZ section stained with LM6 antibodies with detected 1.5 arabinian. We obtained the result was similar to analogous case with antibodies directed against beta-1.4-galactan. We observed minimal fluorochrome emission. The signal emission is higher than in the sample from plants grown under control condition. The emission is visible in most of the observed cells in the form of uniform granulates located in the cell wall. Conclusion. Polygamma-glutamic acid cases change in the chemical structure of the cell wall in the AZ cells of yellow lupin flowers, ultimately leading to the restriction of the upstream process of these organs. Presented results provide novel information for the possibility of use of gamma PJ as a molecule that helps crow to survive under abiotic stress conditions, which is crucial for modern agriculture and development of ecological strategies for plant cultivation. Thank you for your attention. I would like to be delighted to answer any questions from our interested parties. Please direct them to the email address visible on the site. Once again, thank you for your attention and goodbye.
Yes, we also thank you for your presentation, and we have the first question from Dominika Kapuścińska.
Hello, I
Yes,
have
hello.
one question. Do you know or have heard of any other molecules similar to gamma PGA?
Unfortunately, I don't know similar chemical compounds. I have been deeply focused upon understanding gamma PGA nature and potential interactions with plants. I plan to make literature analysis in this direction and another time.
Okay, thank you.
Thank you too.
Okay, we still have some time for a question to our speaker. Yes, we have a question on our chat from Kamil Klinkowski. Do you plan to perform any molecular analysis such as gene expression in your research?
Yes, of course. This is only introduction to more advanced analysis. We focused on microscopic examination and immunochemistry of cell wall components because they are allowed to us quickly and effectively determine gamma PGA supports plants during drought. In future studies, we aim to determine the impact of gamma PGA and antioxidant enzymes. Oxidate stress following drought stress is extremely destructive to because selective oxygens. Additionally, we definitely plan to conduct an analysis of gene expression involved in phytohormone synthesis, phytohormone synthesis, as their interaction and deactivation during the plant treatment of drought stress with gamma PGA.
Okay, thank you. Is there any other questions for our speaker? it's not, it's thank you so much once again for your presentation and for answering the question. And now,
goodbye, have a nice day.
Yeah, have a nice day too. And this was the last presentation of this session. So once again, thank you so much for our speakers. And now I would like to invite you for the extended break. It's about 45 minutes, and we will see each other at 2 p.m. on the fourth presentation session. And I also encourage you to speak with each other on our chat. I will also want to say to Izabela Wysocka that you have a question on our chat, so please could you answer this question?
Hello, can you hear me?
You can answer
okay.
On the chat.
Okay, sorry.
So once again, thank you all and see you on the next presentation session at 2 p.m. Hello and welcome everyone on our last presentation session of the first day of Natural Baltic Science Conference. And at first, at the beginning, we will see the presentation of Jakub Reczkowski from Poznań University of Technology. And the title is Titanium Implant Modified with Zinc Doped Carbon Dots Layer as an innovative coating for the development of local drug delivery system for ciprofloxacin.
Good afternoon, everyone. My name is Jakub Reczkowski, and I would like to present a topic related to the chemical modification of the surface of implants and endoprotheses, which are widely used in stomatology and orthopedic. The topic of our work is titanium implant modified with zinc-doped carbon dots layer as an innovative coating for the development of local drug delivery system for ciprofloxacin. At the beginning of my presentation, I would like to describe some of the main advantages of titanium, which is one of the most popular materials used to produce this type of implants. This metal is well known for its high mechanical strength in relation to its weight, and also has good corrosion resistance. It is used as the main component of alloys for the production of implants and endoprotheses, because it has high biocompatibility and enables cell adhesion on its surface. The most commonly used alloy is the one called titanium 6 aluminum 4 vanadium. It has one significant drawback, which is its susceptibility to the formation of biofilm on its surface. Therefore, the aim of our work was to eliminate this defect by creating an appropriate layer with antibacterial properties on the surface of this alloy. In our work, the tested alloy was covered with a layer of carbon quantum dots doped with zinc cations, which enabled the absorption of the active substance ciprofloxacin, creating a local drug delivery system that eliminates the risk of development of microorganisms on the surface of this alloy. Now, let's move to some properties of carbon quantum dots used to create a layer on the titanium alloy surface. The first important property of this material is that they don't cause any toxicity to human cells in low concentrations. They also have high biocompatibility, and it's caused by their origin, because they are mostly synthesized from endogenic amino acids. Another benefit of carbon quantum dots is the possibility of functionalization of a given surface, and it's caused by the presence of various functional groups in their chemical structure. In this part of my presentation, I would like to point out some disadvantages of the oral administration of ciprofloxacin and why it's needed to find other ways of distributing this active substance. When ciprofloxacin is administered orally, it's quickly eliminated from the body, and it's related to the first pass effect, which takes place in the liver, where enzymes attach hydroxyl groups to the molecule of this antibiotic. Another disadvantage of oral administration of this active substance is the presence of multiple side effects on the digestive system, for example, diarrhea and nausea. I would also like to point out the limited bioavailability, which is also related to the first pass effect. Taking into account the disadvantages of oral administration of ciprofloxacin that I mentioned before, it's necessary to find other ways to distribute this active substance to improve its effectiveness. Going to the next part, I want to say why we decided to use ciprofloxacin to create an antibacterial modified surface of the titanium alloy mentioned before. This active substance, in its structure, has a carbonyl and a hydroxyl group in close proximity, which have numerous lone electron pairs capable of forming coordination bonds with metal cations. Now, I want to focus on preparing our modification of titanium alloy. In the first stage of research, we synthesize a layer of quantum carbon dots by using histidine and citric acid as substrates. The layer was created by immersing titanium plates made of the titanium 6 aluminum 4 vanadium alloy in a solution of carbon quantum dot substrates, after which they were placed in an oven with an appropriate temperature program. In the next stage, titanium plates with carbon quantum dots layer on their surface were immersed in zinc chloride solution for 24 hours to enable sorption of zinc cations. After that, the modified plates were immersed in a ciprofloxacin solution to enable the sorption of its active substance on the created layer. On the screen, you can see the result of the preparation of our layer. That's the sample before modification, and that's the final effect of it. You can see that the surface of the titanium plate is completely covered by carbon quantum dots layer. Now, I would like to focus on the results of our research. The first analysis was taken to determine if the created layer has the ability to sorption of ciprofloxacin. To determine it, we used UV-Vis analysis. You can see the spectrum of ciprofloxacin on the screen while the analysis was in progress. The sorption process lasted 96 hours, and the final amount of ciprofloxacin absorbed on the modified titanium plate was 20.8 micrograms, which is approximately 70% of the initial amount of this active substance present in the solution used for the sorption process. The next analysis, which was scanning electron microscopy, was conducted to check the distribution of the created layer on the surface of the titanium alloy plates after each stage of modifications. We can see that the created layer is not evenly distributed, but there's a significant change if we compare it to the titanium alloy without the modified layer. The presence of a carbon quantum dots layer is proven by these images. In addition to the SEM technique, the EDS analysis was conducted to check the distribution of the characteristic elements that should be present after the occurred modification. In the first row of this image, you can see that after the process of zinc cation sorption, they are evenly distributed on the titanium plate surface. We can also see that in the second row, fluorine, which is present in the ciprofloxacin molecule, is also evenly distributed. It's a crucial feature because all titanium plate is covered by active substance, creating an antibacterial layer on its surface. In the next stage of research, the FTIR analysis was conducted to check the presence of carbon quantum dots on the modified surface of titanium alloy. The bands in the range of 3,500 to 3,000 come from the hydroxyl and amino groups derived from histidine and citric acid used to synthesize carbon quantum dots. In the range between 1,600 to 1,400, we can see the presence of bands coming from carbonyl groups and also from the imidazole ring derived from histidine. Unfortunately, we cannot see any characteristic band derived from the ciprofloxacin because they are covered by bands coming from carbon quantum dots. In addition to the previous analysis, the FTIR mapping was conducted to check the distribution of carbon quantum dots layer on the titanium alloy surface at each stage of modification. We can see that the layer is present and well distributed on every stage of modification. Now I want to focus on the two most important analyses during our research. The first of them is the analysis of the desorption process of active substance from the created layer. The release of ciprofloxacin was analyzed in the simulated body fluid using UVV technique. We can see that the desorption process lasted 4 hours and the total amount of released substance was about 18 micrograms, which is about 89% of the absorbed amount before. Now I would like to move on to the last analysis that was carried out, and it was the analysis of the antimicrobial activity of the modified titanium 6 aluminum 4 vanadium alloy. The obtained results in the table show that our modification of titanium alloy creates a really good material that is resistant to the development of microorganisms. In the first column, we can see that pure titanium alloy enables the microorganisms to grow. The most important thing that comes from these results is the fact that our modified titanium plates stop the formation of biofilm on their surface, especially for gram-negative bacteria in the first two rows. This is caused by the fact that ciprofloxacin is an antibiotic mainly used for the treatment of this type of bacteria. Now, let's move to the conclusions of our research. The modification of the titanium alloy used for the production of endoprotheses and dental implants allows for successful synthesis of a layer of carbon quantum dots on the surface of a titanium 6 aluminum 4 vanadium alloy, which has been done for the first time ever. Elimination of the main disadvantages of this titanium alloy, which is the possibility of biofilm formation on its surface, and creation of an innovative drug delivery system for ciprofloxacin. That brings me to the end of my presentation. If there are any questions, I will be pleased to answer them. Thank you for your attention.
Yes, thank you for your presentation. And as you said, now is the good time to ask some question to our speaker.
Yeah, I'm here with you. I see some questions on the chat.
Question on chat, yes. Yes, so the first question was asked by Kamil Klinkowski. It's under what condition do you conduct ciprofloxacin deposition?
Yeah, I see that. Could you be more precise with that question? Because is that a question related to the EDS analysis or?
Kamil, could you be more precisely?
So okay, maybe I will go to the second. I will go to the second question. Where did the idea of using zinc ions come from? It comes from the research of my supervisor that was mentioned on the first slide, Mariusz Sandomierski. In his researches, he used zinc cations to sort other active substances, and he was doing some researches with the other cations too. For example, magnesium cations and calcium cations. And in every research, the zinc cations were the best to sort the active substances. They are the most capable to sort active substances. So that's the idea why we decided to use zinc cations. I also see the third question. How did you measure the bacteria growth inhibition? To determine this analysis, we used UV-Vis technique. We measured the absorbance, of the growth bacteria or something like that. I don't know how to explain this.
Okay, thank you for your answers. Maybe if there are any other question for our speakers, for example, if Kamil or
maybe I can answer the first because I think that.
Excuse me, can I ask you to answer this question on chat because we have some time schedule and
okay.
it's time to our next presentation. So yeah. Thank you again for your presentation and your answers. And now we will hear the presentation prepared by Natalia Pyra from University of Lisbon. And the title of this presentation is Bioactive Gas Containing Zinc Ions as a Carrier for 6-mercaptopyrene.
Hello, my name is Natalia Pyra, and I will present today the results of my research regarding the drug delivery system for 6-mercaptopyrene. The subject of my work is bioactive glass containing zinc ions as a carrier for 6-mercaptopyrene. It's the drug, cytotoxic drug, which is used for the therapy of childhood acute lymphoblastic leukemia and acute myeloid leukemia. Unfortunately, the drug has its limitations like high drug clearance, low and variable bioavailability, poor absorption, short half-life. This makes it more difficult to achieve the full therapeutic effect when taken orally. The oral drug delivery system, among others here, which can be seen in the figure, belongs to the conventional system. This is not enough to overcome the disadvantages of 6-mercaptopyrene. Therefore, there is a need to develop modified drug delivery systems. And bioactive glass containing zinc ions can be such a modified drug delivery system for 6-mercaptopyrene. Here, briefly, the aim of the work First, synthesis of bioactive glass, then doping the material with zinc ions, next characterization of the morphology and surface of the obtained particles, and the most important, sorption capacity test and drug profile study. Here in the figure, we can see the mechanism of drug sorption on the surface of mesoporous material or either in its pores. The drug attaches to the molecule by forming coordination bond between the sulfur atom, which is derived from the structure of the drug, and the zinc ions with which the material is soaked. To characterize the morphology of synthesized particles, scanning electron microscopy analysis was performed. Figure 2 presents the materials before and after drug sorptions. Both variants do not differ significantly from themselves. The result could indicate drug adsorption either on the surface of the bioactive glass nanoparticle with a monolayer pattern or either inside the pores. No other structures appeared after the drug absorption. What proves the no transformation of 6-mercaptopurine carrier into other compounds? Images show mostly oval particle morphology with a uniform size distribution. However, some particles have an elongated oval shape. This may be due to the method of synthesis, which can be improved a bit in the future, or the choice of apparatus. The size of obtained particles was determined using ImageJ program. The results of this analysis are shown here. Here we have, like, for the sample before sorption 339 and after 338. Those values don't differ significantly from each other. This indicates that the sorption process took place without the formation of new structures. This analysis provides the information about chemical composition of material before and after drug sorption. Here we have studied 3 elements: silicon, zinc, and sulfur. Silicon because it's predominant element of bioglass structure, zinc because the material is doped with zinc ions, and sulfur because it stands for the drug. All right, so zinc ions are distributed in the same pattern as silicon ions. It indicates that the sample is doped with zinc ions equally throughout the material, which allow 6-mercaptopurine to bond. The more homogeneously zinc ions are distributed, the more equally drug is retained. And that's important because consequently it provides the delivery of 6-mercaptopurine in the same doses from every volume of material. However, it will be later justified in the release profile. Moreover, this analysis confirmed the presence of sulfur. Of course, it is one of the elements that is after drug absorption, because it is one of the elements that is contributed to the structure of 6-mercaptopurine, and that proves the effectiveness of drug sorption. The chemical structure of the synthesized material before and after drug sorption was analyzed by FDIR. The results clearly show that the synthesis of bioactive glass with zinc ions was carried out successfully. For example, this broad band is attributed to asymmetric stretching vibrations of bonds between silicon and oxygen. New bands occur after sorption of the drug. For example, the smaller band here can be attributed to bending vibration of the NH bonds. And for example, here the smaller band may characterize the stretching vibrations of NH. Okay, the distribution of 6-mercaptopurine on the surface of the bioactive glass nanoparticles was determined by FTIR imaging, and the analysis parameters were set in such a way that the blue color here stands for the absence of the drug. The color change in the imaging spectra indicates the presence of a certain amount of the drug on the surface in the bioactive glass particles. Therefore, it can be observed that area regarding the sample before drug sorption is blue, showing that the carrier is not coated with drug. The sample after sorption is almost completely covered with drug, as evidenced by the colors indicating the amount of the drug occurred at the given measurement points. Overall, the test confirms the effectiveness of drug retention on the carrier. And here, particle size, zeta potential. The particle size of bioactive glass before and after drug sorption was also performed, determined by performing DLS analysis. Zeta potential was examined to determine the material's susceptibility to agglomeration. And here we have values obtained for zeta potential. Based on those, it can be seen that bioactive glass nanoparticle before 6-mercaptopurine sorption have a slightly higher tendency to agglomerate than the drug-coated carrier. And the results are similar to each other, with both samples in the range of absolute values from 10 to 30 millivolts. This indicates an initial instability. This is a moderate tendency to agglomerate. The diameters of the bioactive glass particles before drug sorption are 1040 and after 1057 nanometers, and the size of the obtained particles is much larger than the size obtained previously using ImageJ software. That may be due to the agglomeration. And finally, sorption of mercaptopurine. UV analysis confirmed the material's ability to sorb the drug, and this amount was retained, 313 micrograms. The desorption process was monitored using two different pH, 7.4 and 5 respectively. This range helps explain how pH stimulates the release of the drug going from the physiological fluid environment to the slightly acidic environment of the malignant cells. In the case of 6-mercaptopurine, it's important for the carrier to ensure prolonged release of the drug in the body, as it's designed to work against short half-life of the active ingredient. The drug release profile under both neutral and acidic conditions is shown in the figure. Controlled release at pH 7.4 lasts 25 hours. The values of released drug after each hour of the conducted test are similar, but becoming smaller, of course. In contrast, release in acidic environment ends after 75 minutes. As expected, the process takes longer under neutral conditions, and the released dose of 6-mercaptopurine represents about 53% of total amount of drug absorbed in the carrier. This indicates that the carrier is highly stable in the neutral environment of body fluids, and the carrier has the potential to overcome the main limitations of 6-mercaptopurine as drug, which can be used now in targeted anti-cancer therapy. And bioactive glass doped with zinc ions can definitely increase the therapeutic effect and provide greater comfort and safety for the patients. And here we have finally conclusions. and synthesis of the carrier for 6-mercaptopurine was successful. The carrier has the ability to solve the drug, and controlled release of the drug was achieved. Thank you so much for your attention, and now is the time for questions if you have.
Yes, thank you for your presentation. And I see the question from Dominika Kopuścińska.
Yes, hello. I have one small question. What form and way the drug-modified nanoparticle could, in theory, be served to the patient?
Hi, in general, the carrier is in the form of powder. So the main idea is to for the drug on the carrier to be injected, like intravenously. So yeah.
Okay, thank you.
Yes, thank you for the answer. Is there any other question to our speaker? Okay, if it's not, thank you again for your presentation, Natalia. And now I would like to invite everyone for the presentation of Julia Chrzan from Krakow University of Technology. And the presentation title is Microwave Synthesis of New Anilinotriazine and Derivatives with Potential Biology Activities. However, Julia can be with us today, so this presentation will be without question after it. But if you have any question, I encourage you to write them, and we will send those questions to Julia via email.
Good morning. My name is Julia Chrzan. I'm a master's student in chemical technology at the Krakow University of Technology. I'm very sorry that I'm not able to participate in the live conference, but I would like to present you some of my research, which is also my diploma thesis. The topic of my work was the microwave synthesis of new anilinotriazine derivatives with potential biological activity. The aim of my work was the synthesis of new anilinotriazine analogs with potential biological activity using microwave radiation. The range of the diploma thesis was a literature study in the field of microwave methods of synthesis and biological activity of anilinotriazines analogs. A synthesis of selected compounds in the microwave radiation field, and cleaning and confirming the structure of the obtained connections. At the very beginning, I would like to present in figure number 1 the rank of the 1,3,5 triazine molecule, which is a fundamental molecule in my research. It has cosmetic properties as a basis for sunscreens and therapeutic properties, e.g. antibacterial, antifungal, anti-malarial, antiviral, anti-inflammatory, anti-tuberculosis, and anti-cancer, which are the ones I focused on the most. The triazine molecule has been tested on colon cancer, breast cancer, prostate cancer, lung cancer, and ovarian cancer cells. However, I became interested in the topic of triple-negative breast cancer, which is by far the most malignant form of breast cancer. The mortality rate and the predispositions to recurrence of this disease are enormous. In the next slides, I will discuss the methodology of my research, synthesis, and condition. So the synthesis consists of 4 steps. In stages 1 to 3, for each chlorine, I connected the structures I was interested in different conditions. In the third stage, I also decided to optimize the conditions of the microwave synthesis of the last stage, where after finding the most favorable conditions, I could add other compounds using the last chlorine in the fourth stage. In the case of my work, that was amines. So the first stage was carried out under conventional conditions at reduced temperature. 2-chloroaniline was attached to the 1,3,5-triazine molecule. The reaction took place using the isopropylamine as a base and tetrahydrofuran as a solvent. Similarly to the first stage, the second stage was also carried out under conventional conditions at a reduced temperature, and after the introduction all of substrates, the mixture was brought to room temperature. Morpholine was attached to molecule number 1 using the same base and solvents as in the first step, so that was the isopropylamine as a base and tetrahydrofuran as a solvent. In the third stage, as I mentioned at the beginning, optimization of the synthesis conditions was carried out, where 6 different bases, 5 different phase transfer catalysts, and 2 different solvents were tested. The reactions took place in microwave conditions with a power of 50 watts and a temperature of 150 degrees Celsius and for 150 seconds. The compound on which the optimization was carried out was 2-phenylethylamine. The tables contain exactly the substances that I tested in stage 3. The most favorable conditions turn out to be sodium carbonate as a base, tetrabutyl ammonium bromide as a phase transfer catalyst, and dimethylformamide as a solvent. In the fourth stage, I added the remaining amines of my choice, aniline, benzylamine, and 3-phenylpropylamine. The reactions proceeded with high or medium yield. Compound number 3 was tested for cytotoxicity in the MTT test on 3 triple-negative breast cancer cell lines and 1 healthy line. Comparing the cytotoxicity of compound number 3 and cisplatin, which is used in current therapies against this type of breast cancer, it can be concluded that my compound is moderately or weakly cytotoxic, but doesn't show any toxicity towards the healthy line. In silico screening against early ADMET parameters, it can be seen that the tested compound meets Lipinski's rule of 5, which makes it drug-like. During and after my research, I managed to draw some important conclusions, primarily that the compounds number 3, 4, 5, and 6 I obtained, which are analogs of 1,3,5-triazine, are new compounds unknown in the literature, and that compound number 3, despite its weak activity, was not toxic to a normal cell line, which is important in targeted therapy, the aim of which is to destroy cancer cells while sparing healthy cells. The tested group of compounds requires further chemical biological work to improve AG cytotoxicity. As I said earlier, the most favorable conditions for obtaining model compound 3 are sodium carbonate as a base, TBAB as a PT catalyst, and DMF as a solvent. I also present to you the literature I used when writing my thesis. And thank you very, very much for your attention. If you would like to ask me a question that I've unfortunately unable to answer today, please contact me by email at the address I provided on the slide. So once again, thank you very much.
Yes, we are glad that we could hear the presentation of Julia Chrzan. However, that's what I said and Julia said. We can ask her any questions. So, but if someone wants to ask, you can also write on our chat and we send it to Julia. So now it's time to our last presentation of this session, and this presentation will be presented. This video was recorded by Katarzyna Staroń from Krakow University of Technology, and the title of this presentation is Ultrasonic Synthesis of New Benzyltriazine Derivatives with Potential Biological Activity.
Good morning, everyone. The subject of my presentation today is ultrasonic synthesis of new benzyltriazine derivatives with potential biological activity. Cancers are one of the larger and more serious groups of disease of civilization which can originate in almost any organ or tissue of the body. Their development is a complex process that affects regardless of age and health status. Children, adolescents, as well as adults are exposed. Everyone's lifestyle plays a crucial role in cancer prevention. Cancer is determined by external and microbial factors, and its etiology is multifactorial. Triazine analogs are used as a therapeutic agents due to their potent antiviral, antimicrobial, antimalarial, anti-cancer activities. Triazines are also characterized by their ability to absorb a broad spectrum of UV radiation, UVA and UVB, antioxidant activity, and thermostability. The research focuses on obtaining new 1,3,5-triazine derivatives with potential biological activity against colorectal cancer cell lines. The current literature on cancer characterization and application of 1,3,5-triazines in anti-cancer therapy were reviewed. The study was divided into steps: reaction conditions, products purification methods were described. Taking into account the review of previous literature data and indicating the attractiveness of unconventional synthesis methods, this work has just proposed a variant for conducting synthesis in the presence of ultrasound. With this method, the reaction running time can be significantly reduced, while high yields can be achieved. The first two stages of the reaction, which do not require the provision of energy, were carried out as standard using flasks and steering on a magnetic stirrer. The first reaction was carried out at a temperature not exceeding 0 degrees Celsius for 2 hours, while the second reaction was carried out at room temperature for 24 hours. The third stage of the reaction, which required a supply of energy, was performed using an ultrasonic reactor. Monitoring the progress of the reaction was carried out using the thin layer chromatography, TLC method. The purity of each product obtained was determined using ultra-performance liquid chromatography, mass spectrometry, UPLCMS. In the first step, benzylamine was mixed with cyanuric chloride and DIPA, and THF was used as a solvent. The reaction yielded compound number 1, which was purified by extraction with dichloromethane, water, and the addition of hydrochloric acid. The reaction yield was 59%, and the purity of UPLCMS was an indication of more than 90%. The second step used the product from step 1. The product of reaction 1 was mixed with ammonia water and THF was used as a solvent. The product of this reaction was compound number 2, which was purified by extraction with dichloromethane, water, and the addition of hydrochloric acid. The yield of the reaction was more than 89%, and the purity of UPLCMS was an indication of more than 96%. A series of test reactions were performed, which resulted in the final product being condensed with an excess of 2-phenylethylamine to obtain product number 3. This step involved evaluating the effect of the presence of various bases, sodium carbonate, potassium carbonate, sodium hydroxide, and 2 solvents, water and DMF, on the yield of the final product. TBAB was used as the PTC catalyst. Each reaction was carried out in the presence of ultrasound in a reactor for 5 minutes. A series of test reactions were carried out with a strong inorganic base, sodium hydroxide, and weak bases, potassium carbonate and sodium carbonate. The most favorable base in ultrasonic reactions using water as a solvent was sodium hydroxide due to its high reaction yield, which was 79.4% 3C, and the least favorable was sodium carbonate 3B, for which the yield was 31.5%. In contrast, in synthesis using DMF as a solvent, the most favorable base was sodium carbonate 3E, where the yield was 88.5%. For the synthesis carried out in the presence of sodium hydroxide 3F, the lowest yield was 35.3% was obtained. A ultrasonic reactor can significantly contribute to reducing the time of the synthesis carried out. In addition, the minimalization of energy and solvent consumption makes the procedure compatible with the concept of green chemistry. Intermediates were obtained in 59% for compound number 1 and 89.4% for compound number 2. Yields and had high UPLCMS purity for compound number 1 over 90%, for compound number 2 over 96%. The reaction profile composition of the post-reaction mixture according to TLC for 3A to 3D products using water as a solvent was found to be cleaner compared to the reaction profile for 3D to 3F products using DMF. The best base for reactions carried out in water is sodium hydroxide, and at 79.4%, and for reactions carried out in DMF is sodium carbonate at 88.5%. That's it today. Thank you everyone for your attention.
Yes, thank you for your presentation, and now it's time to ask the question to our speaker. But taking the opportunity to speak, maybe I will ask the first question. There was a big problem with the non-selective products of substitution. on this on those steps of synthesis?
Good morning, everyone. I'm sorry, but I have a technical problem with my computer, so far no, I am. Please, can you repeat?
Yes, I have a question. Do you have the problem with non-selective substitution on some kind of steps of your synthesis?
Do I have a problem?
Okay. Do you hear us, Katarzyna?
I'm sorry, please, can you write in chat, because I don't hear you.
Okay, so this is definitely okay. My question may be the second or third. We have a question on chat from Daria Wada. Do you plan to perform tests to check the effectiveness of these compounds on living organisms? If so, what types of analysis?
Yes, I will testing the effectiveness of these compounds on living organism on the mouse, I guess. Cancers are a huge problem, the most important civilization disease, and I guess trazine analog will be the solution for cancer cell lines in therapy.
Okay, thank you. I see the question from Katarzyna Wiktorska.
Hello again. Concerning the question about biological tests, I would advise to these compounds on the cell lines before you go to check it on the living organism. So maybe you have performed such tests in vitro, not in vivo.
Yeah, I will check in vitro and also in vivo, but this is the next steps. This is my beginning, and I want to try each option.
Okay, so good luck.
Okay, thank you.
Thank you.
Okay, is there other question to our speaker? Okay, so maybe I will ask the another question. What is the cost of this synthesis using the ultrasonic method instead of classical reaction?
How much is cost? Yeah.
Yes.
Cost is very low. This is the time also is low, and I try to do my work under the green chemistry. So
I'm sorry, Katarzyna, but we are not hearing you. Okay, we have some technical issue with answer the question, so maybe it would be good to answer the question on our chat. So if there are any other question to the speaker, so please write them on the chat. And now we heard the last presentation of today, which means that we complete the 4th session of the presentations. However, this is not the end of our highlights. I would like to invite you to a break during which you can enjoy the recording of our poster presentations. Afterward, the poster session will begin, during which you will be able to ask questions of poster authors. And I also have some technical remarks. To access the recordings, you need to go through the website, our website, click on the Join Poster Session button, select first day, and the password of those posters is NSBC 2024. So now I think we could start the break.